Good morning, good afternoon, good night. Welcome to everyone. I have to say that this is a great privilege for me to do the chairmanship of this fourth World Congress on bronchiectasis and on non-tuberculous mycobacteria. I want to thank all attendees, almost 400, I think, according to my last news. I want to thank really very warmly all the speakers for the effort that they have done and the pharmaceutical industries for supporting this Congress. And I'll do some additional thanks at the end. My second point now in this welcome address is to talk a little bit about bronchiectasis. Uh, it's amazing. It's amazing how in this pandemic, in this year of living dangerously, now we have the opportunity to do something in bronchiectasis, which is has been a kind of neglected respiratory disease. And it's amazing to be here reviewing all new things, all new scientific information in bronchiectasis. So I am really, really, really very happy and very proud to be here and to see that bronchiectasis is going ahead. But I cannot forget COVID for several reasons, because I feel this in my heart. I have a lot of respect, and I ask a lot of respect for professionals that have been in front line first, professionals, nurses, clinicians, all the personnel. Second, scientific community. It is amazing how the scientific committee has reactionated, the reaction of scientific community, more than 47,000 papers in 1 year, is what I said yesterday. Unbelievable. And thank as well to the pharmaceutical companies, because I think that they made a lot of effort putting everything together, designing trials, vaccines, etc. So I cannot forget COVID. I cannot forget bronchiectasis. And my final thanks are for public relations, especially for Marcia Folergani and all her team that allowed that this Congress went ahead, because this Congress was planned at the beginning online, be in vivo here in Barcelona. We changed the dates, and finally we decided to do this virtually, which is not an easy task. Thanks, Marcia. So I hope that you will enjoy the Congress. I thought that you will learn a lot. And thanks again for being with us this year, now from Barcelona, but from every place of the world. I'm passing the word to my dear friend, Francesco Blasi, and later on to James Chalmers, another very good friend. Francesco, please.
Thank you, Tony. It's my pleasure also to give the address of the co-chairman in this case. This is the fourth edition of the World Bronchiectasis Conference, and we add also the idea that NTM are also important in bronchiectasis. So we move a little bit forward our conference. We started some years ago in 2016 with the Hanover meeting. It was the first World Conference on bronchiectasis. And I think we had made a lot of big steps in understanding bronchiectasis. And if you look to the number of papers that are appeared in the literature on bronchiectasis after the World Congress in 2016 in Hanover, certainly bronchiectasis is now a really hot topic in the literature. And this is due mainly to the effort of many people that start to study bronchiectasis. After a while, when the diagnosis certainly dates back many years ago, but the definition. But now we are really making a step forward in the understanding of bronchiectasis and an understanding all the comorbidities that are around bronchiectasis. And I think after the Milan conference in 2017 and the Washington conference in 2018, now we are virtual. I think all of us are a little bit upset that the fact we are not face-to-face in the fantastic city of Barcelona, but still want to work together and make a further step in the understanding of bronchiectasis and NTM infection in this virtual edition of the Congress. As Tony said, we are almost 400 participants today, and from 40 nationality. And this is important. It's a real world Congress. From Brazil to New Zealand. We have people coming from all the world. I must say that Spain and UK are the two leading countries in terms of participants. This is important. We have more than 70 abstracts submitted. We have 2 rising stars, but we will have the opportunity to hear them just after this introduction. And we have 5 oral presentations that are being awarded, and 60 posters presented. So I think it will be a very live conference. And I hope really, as Tony Torres said, that you will enjoy really this virtual edition, and hoping that in the next year we can have the opportunity to have a face-to-face meeting that is still so important, to see each other and work together, and certainly is important. And with this, I thank you very much, all of you, and I pass the word to James Chalmers, that will give his address on behalf of Embark.
Hello, my name is James Chalmers, and it's my pleasure to welcome you on behalf of Embark to the 4th World Bronchiectasis and NTM Conference. Ever since our first conference in Hanover, the World Bronchiectasis Conference has been such an important part of the development of the Embark project, and also of developing clinical research and clinical care in bronchiectasis. It's wonderful to look back over the last 5 years at the progress that we've made in this disease, the progress that we've made in research, and just in raising the profile of this disease across respiratory medicine. And I think the conferences play an important part in that, because it brings together groups of researchers to talk about important new data, and it also brings together and brings through young investigators who are really driving this field forward. I think the conference this year is very special because we've managed to pull this together. We've managed to all come together despite all the challenges of COVID-19, and to show our commitment to keep working on new therapies and new ways of treating bronchiectasis. I hope you enjoy the next few days. I hope you enjoy the conference, and I look forward to seeing you in person for the 5th World Bronchiectasis Conference and beyond. Thank you.
Francesco, please.
I think, Tony, you had to introduce the first speaker.
Okay, thank you.
Sorry.
Excuse me?
Just an introduction. It's my pleasure to introduce the new session on the Rising Star session. You know that in our Congress, in our World Congress, we have always the possibility to hear 2 rising stars that they are awarded with the possibility to present in a plenary session their own work that had been evaluated by the steering committee as the 2 best possible presentation and the research that had been submitted to the evaluation of the steering committee. So it is my pleasure to give the floor to Tony Torres to introduce the first speaker.
Thank you, Francesco. So one of the rising star is Holly Kerr from Dundee, United Kingdom. She's going to present a sputum proteomics identifies mechanism of disease severity and treatment response in bronchiectasis. We will have 5 minutes for discussion after her talk. And you can send, the audience can send questions that we will try to forward to the speaker. Holly, please, and congratulations. You can start, Holly.
Thank you for that introduction. I'm hoping you can see my slides.
Yes.
My name is Holly Kerr and I'm a final year PhD student at the University of Dundee. As we all know, bronchiectasis is poorly understood, but neutrophilic inflammation is believed to be central to its pathophysiology. Over the next 10 minutes, I'm going to take you through a series of UK and international studies that we've used to investigate the underlying mechanisms of neutrophilic inflammation. And hopefully I'm going to demonstrate that neutrophil extracellular traps, or NETs, a form of antimicrobial defense, are implicated in disease severity and can be reduced through treatments such as macrolides and antibiotics. Our initial study used liquid chromatography mass spec to compare protein profiles in sputum between severe and mild bronchiectasis patients, with 20 in each group. The PCA plot on the left-hand side shows individual patient protein profiles, with mild patients shown in green and severe patients shown in red. And what we found was there was two clusters present. On the right-hand side, you can see a more mild cluster in the blue rectangle. On the left-hand side, there was a cluster of more severe patients in the orange rectangle. When we look at the loadings plot on the right-hand side, we can see what proteins are driving these clusters. In the more mild group, this was characterized by antiproteases and endothelial proteins, but the most divergent and discriminating proteins were found in the more severe group. Severe disease was characterized by the upregulation of neutrophil and neutrophil extracellular trap proteins, such as resistin, elastase, MPO, and azurocidin 1. So what are neutrophil extracellular traps, or NETs? NETs are a form of neutrophil antimicrobial defense, and they're able to bind and trap and kill certain pathogens. However, they've also been demonstrated to cause local and systemic damage to lung tissue. NETs are web-like structures composed of chromatin and over 30 different granular proteins. And in the following studies, we've measured NETs in sputum using a validated histone H1 elastase immunoassay, which we developed here in Dundee. So to explore that relationship between NETs and bronchiectasis, we first used a UK observational cohort study of 175 patients who were recruited here in Dundee. We found that there was a significant correlation between bronchiectasis severity and increasing NET concentrations in sputum, and that patients with higher NET levels were significantly more likely to have severe exacerbations. Additionally, patients with high NET levels reported a loss of quality of life as measured by the Colby score. To further understand this relationship, we split our NET concentrations into 3 even tertiles: a low tertile shown in red, an intermediate tertile shown in green, and a high tertile shown in blue. And what we found was that patients in the highest tertile had the shortest time to next exacerbation and a significant increase in mortality. This data begins to suggest that NETs are associated with disease severity in bronchiectasis. We also looked at the microbiome profiles of these 175 patients using 16S rRNA sequencing. We initially looked at alpha diversity, as seen in the graph on the left-hand side, and again we looked at those 3 NET tertiles, with the highest NET tertile being shown in blue. What we found was patients with the highest level of NETs had the greatest loss of alpha diversity as measured by both the Shannon Immunodiversity Index and the Cow Index. When we looked at microbiome clusters as a whole, when we looked across the 3 tertiles, we found that the 3 tertiles all had significantly different microbiome profiles from each other, and they really clustered together. And you can see that in the blue oval shape is our high cluster. Severity is often driven by a loss of commensal taxa and a dominance of the single taxa. And we can measure this form of dysbiosis by using the Berger-Parker index, which is a measure of dominance. And what we found was, again, patients in that high tertile shown in blue had the highest Berger-Parker index, so that overgrowth of 1 taxa. We were interested to know what pathogens are causing this. And when we looked, we found that it was Pseudomonas and Haemophilus, the proteobacteria, that were most highly associated with high NET levels. This data suggests that NETs were associated with loss of microbial diversity and an increased relative abundance of pathogenic proteobacteria. Next, we sought to verify and validate the findings of the UK cohort in a much larger European study. And for this, we used the Bridge European Observational Study of 275 patients who were recruited across 3 centres in Europe: Dundee in the UK, Milan, and Barcelona. The results of this study were concordant with the previous smaller UK study. Again, we saw that NETs correlated with an increase in severity and a reduction in col-B. We saw that patients with severe exacerbations had higher NET levels. And for this group, we also had FEV1 data. And we were able to see that patients with increasing NET levels had a reduction in lung function as measured by FEV1. The data from these two cohorts suggests that NETs are associated with an increased relative abundance of pathogenic proteobacteria and are upregulated in severe disease. We therefore investigated sputum protein profiles in response to 14 days of antibiotic treatment in patients with exacerbations. For this study, we used label-free liquid chromatography mass spec to identify proteins associated with treatment response using paired samples taken during an acute exacerbation. We took samples on day 1 of the exacerbation and again on day 14 after 2 weeks of antibiotic treatment. And we had 20 patients in the first group. The results showed that antibiotic treatment significantly altered the expression of 39 sputum proteins, with the neutrophil degranulation pathway being the most strongly implicated in response. The volcano plot on the left-hand side shows the differential expressions of sputum proteins between day 1 and again on day 14 after antibiotic treatment. On the right-hand side in blue, we can see the proteins that were upregulated, and these were a mix of proteins, including mostly antiproteases. On the left-hand side in red, we can see the proteins that were downregulated on day 14 after antibiotic treatment. Among the 11 downregulated proteins related to the neutrophil degranulation pathway, 10 were known to be NET-associated proteins, with the 11th being known to be released upon the activation of neutrophils by extracellular chromatin. Additionally, we found that patients who were Pseudomonas positive on day 1 had higher baseline levels of NET proteins such as MPO, elastase, and azurocidin-1, and lower levels of several proteins, including protease inhibitors such as SLP. There was a strong trend showing that patients with Pseudomonas infection had a lower overall magnitude of reduction in their NET proteins compared to those without Pseudomonas infection on day 1. We validated these findings in a second study with the same design, so again 20 patients. Of the additional 20 patients studied, the graph on the right-hand side shows NET concentrations on day 1 and day 14, and we found that 19 out of 20 patients showed a reduction in NET concentration in sputum after antibiotic treatment. However, as you can see in green, these patients are the patients who were Pseudomonas positive on day 1, and they had a blunted response and a lesser reduction in their NET proteins compared to patients who were Pseudomonas negative. Additionally, we saw that patients who were Pseudomonas positive on day 1 also had a shorter time to next exacerbation. In both studies, Pseudomonas negative patients had a greater improvement in their clinical outcomes compared to patients who were Pseudomonas positive. Our results so far suggest that disease severity is associated with neutrophilic cellular traps and that NET levels can be reduced and clinical outcomes improved with antibiotic treatment. However, our data has also shown that systemic antibiotics have a lesser effect on NETs in the sputum proteome in patients who are Pseudomonas positive. So what treatments would be suitable for these patients? Long-term macrolides are regarded as not having antimicrobial efficacy against Pseudomonas, and in a previous study, they have been shown to not change the microbiome in patients who are Pseudomonas positive. However, in vitro, they have been shown to inhibit NET formation. The graph on the left-hand side shows pooled data from prior randomized trials that shows that macrolide treatment prolonged first time to exacerbation. We therefore hypothesized that macrolides would be effective in reducing NETs in a Pseudomonas-infected patients. For this, we looked at 2 studies of long-term azithromycin, one in bronchiectasis and a post-hoc analysis of the AMAZES trial in asthma to investigate the effect of macrolide treatment on NETs. The graph on the left-hand side shows that macrolide treatment for 12 months in Pseudomonas-infected patients with bronchiectasis resulted in a statistically significant reduction in NETs, which was not observed in individuals not treated with macrolides. Additionally, macrolides have established efficacy in COPD and severe asthma where NETs are implicated. To validate and extend our observation that macrolides reduce sputum NETs, we studied patients from the AMAZES trial of long-term azithromycin in asthma. In this controlled trial, 12 months of treatment with azithromycin significantly reduced sputum NETs. To conclude, the study included 538 patients across a series of UK and international cohorts to identify and validate biomarkers associated with disease severity in bronchiectasis. The presence of NETs is associated with the burden of disease, and that treatment response is linked to success with reducing NET levels of intravenous antibiotic or macrolide therapies. Our data suggests that targeting NETosis with immune-modulating therapy is likely to have clinical patients in patients with bronchiectasis, and also may reduce a broad range of neutrophil and downstream inflammatory mediators without compromising bacterial clearance. So what does this mean for current or future treatments? A recent randomized study of DPP-1 inhibition in bronchiectasis showed for the first time the benefit of directly targeting neutrophilic inflammation. Inhibition of DPP-1 in the Willis study was shown to prolong the first time to exacerbation in a phase 2 randomized placebo-controlled trial. Intracellular neutrophil elastase triggers NET formation through the degradation of F-actin and histone H4 and is prevented through the inhibition of DPP1. Our results here may provide a potential mechanistic basis for the beneficial effects of DPP1 inhibition in bronchiectasis. We had a huge number of collaborators who worked on this study, so I'd just like to extend our thanks to our co-authors in Spain, Italy, Belfast, and Australia, and of course my co-authors in Dundee, especially Professor Chalmers, Dr. Schumark, and Dr. Huang. I'm aware I've just covered a lot of data in the last 10 minutes, but if you are interested in reading any more of this, we've just had it accepted for publication in the Lancet Respiratory Medicine. Thank you.
So thank you very much for your very, very interesting presentation. I think that, I don't know if I have questions from the audience, but I have a questions for you is the following. Given your results, first, was Pseudomonas aeruginosa the most frequent, both in microbiome and standard cultures, isolated microorganisms in exacerbations? And then, if you have seen effects of macrolides, do you think that should we give macrolides at exacerbation in patients with Pseudomonas and bronchiectasis, of course?
So to answer your first question, I think out of the 20 patients in the initial exacerbation study, 11 of them were Pseudomonas positive. For your second question, whether or not macrolides are an appropriate treatment, I think I'm not a clinician, so I don't prescribe macrolides to patients, but from our data, it would suggest that although macrolides don't affect the microbiome and they don't have activity against Pseudomonas directly, they do seem to have an immunomodulatory effect against NETs themselves. And we've seen from both asthma and bronchiectasis now that if you treat with macrolides and you reduce NETs, you do improve patient outcomes. So I think this would make treating patients with Pseudomonas with macrolides maybe more appropriate.
During exacerbations, eh?
During exacerbations, yeah.
So this is different compared to the stable period. Francesco, do you have a question?
Well, I was part of the paper, so
okay.
It's difficult to ask anything. Just to, you can just give us some ideas of the possible limitation of the study, just to, in terms of methodology applied or so on, just to give an idea that you think also to other possibility to go in depth in the understanding of the problem.
So I guess we used sputum for everything, which is always, I know everyone has a favorite for microbiome. If they prefer Ballor, they use sputum. We use sputum because, this is such a large number of patients. We couldn't have done bronchoscopy on 538 patients, but sputum does come with its own limitations. It does have saliva contamination. Quality can affect what you get back. And it doesn't include always the lower airways. So that would be a limitation. But I think on balance, we evened it out with the number of patients we saw and the fact that we were able to replicate the data over multiple cohorts.
Holly, I have a question from the audience, from Michal, it's not Michael, Michal Steinberg. Hi, great work. In the pre-post antibiotic cohort, did NET proteome level reduction post-antibiotic course correlate to a reduction in bacterial load?
So yes, antibiotic treatment did reduce bacterial load. I think we measured that in the second cohort. But it was really the thing that was important was the clinical characteristics. Patients who had their greatest reduction in NET levels had the biggest improvement in clinical symptoms, but the patients with the lower reduction in NET levels, those Pseudomonas positive patients, had a less improvement in their clinical symptoms.
So thank you very much. If there is not, there are not more. No, it's the same. Sorry. No, Ricardo, another from Ricardo Jose. How do you know that NETs are the issue driving pathogenesis rather than just an outcome? And between brackets, a biomarker of inflammation, e.g. like C-reactive protein?
So that's a great question. I think part of it would be that there are things in the literature that show that NETs cause local and systemic damage to lung tissue. In CF, it's quite widely accepted that NETs damage the lung tissue and cause problems. I think the elastase would be a good example. So studies of elastase inhibitors have been done and haven't been shown to improve patient outcomes. But if we've shown here, hopefully, that if you can block NETs, you do improve patient outcomes, which would show it's not just a biomarker, but they are causing some kind of damage.
So thank you very much, Holly. Congratulations again. Francesco, can you introduce the second speaker, our second rising star?
Perfect. Great lecture, really great lecture. Thank you. And now it's time to introduce Jody Schildkraut. And the presentation will be on dual RNA-seq of Mycobacterium avium, identifies characteristics of pathogen interactions. Please, you can share your screen. Excellent. Thank you.
Is everyone seeing my screen now? Or?
Yes.
Yes.
Okay.
Okay, thank you for the nice introduction. So as just mentioned, I'll be talking about my work on dual RNA sequencing of Mycobacterium-infected macrophages. So I'm sure, as you all well know, Mycobacterium avium is an opportunistic pathogen. It's actually one of the most common causes of NTM pulmonary disease. And although it's widespread in the environment, the majority of individuals that encounter these Mycobacterium avium will never develop disease. And although many predisposing factors such as underlying lung pathology, immunodeficiencies are known, many other factors that contribute to disease progression still remain unknown. And in addition to that, as we just heard in Holly's talk, clarithromycin, which is one of the most important drugs in NTM treatment, is actually also used as an immunomodulatory drug in at-risk patients. But the effect of this on the interplay between the host and the bacteria is not actually known. So in this study, we wanted to use dual RNA sequencing to study this interplay between host and pathogen, and also the effect of clarithromycin. So while traditional RNA sequencing studies actually analyze the transcriptome of either the pathogen or the host, dual RNA sequencing allows us to study changes in both simultaneously. So going into this study, we had two very specific research questions we wanted to answer. So the first was, what are the interactions between the mycobacteria and the infected macrophage? And the second being, how does the intracellular environment actually influence the bacterial response to clarithromycin? So at our center, we're lucky to have an up and running method of isolating mononuclear cells from healthy donor blood and then differentiating them into macrophages. But because of this dual RNA-seq methodology, we needed a really large amount of biomass to be able to sequence the bacteria deep enough to look at the transcriptome. So first we needed to know what the maximum amount of mature macrophages was so that we could isolate from one single individual's blood. And we found that to be around 50 million mature macrophages, infectious type macrophages. And because of that, we established the following infection protocol. So on day 0, we isolated peripheral blood mononuclear cells from about 500 milliliters of donor blood. This was then differentiated into macrophages over the course of about 7 days using growth stimulating factors. And then after 7 days, the mycobacteria were infected with, the macrophages were infected with mycobacteria at a multiplicity of infection of 5, which we had previously determined to be the most optimal to get the highest ratio of bacteria to macrophage. Which is important to be able to get enough bacterial reads. So we infected for about 24 hours. And then after that, we washed the cells extensively to ensure that all the extracellular bacteria had been removed. And we were really only sequencing bacteria that had been intracellular at the time of isolation. And then we did a total RNA isolation in which we also enriched for the bacterial RNA. Using these methods, we were able to get at least 4 conditions from each donor. So we had 4 conditions of 10 million human cells infected, a multiplicity of infection of 5. And we came to the following experimental setup because we wanted to include at least 3 biological replicates to ensure that all biological variation, particularly variation between donors, had been captured. So from each donor, we isolated 4 separate sets of macrophages, of which half were infected and half were not. So we could make the comparison between uninfected and infected macrophages. And then we also included a Mycobacterium avium condition in which the bacteria were grown in the absence of macrophages, so we could compare the extracellular and the intracellular bacteria. In addition to this, to study the effect of clarithromycin, to one of each condition, clarithromycin was added at 1 microgram per milliliter. So in this talk, I'm going to focus mostly on the bacterial response. I did want to shortly show the transcriptomic changes in the macrophage as well after encountering the mycobacteria. So in human conditions, we saw between 900 and 1,500 differentially expressed genes, which we used to do gene ontology enrichment analysis. And as expected, following the exposure to Mycobacterium avium, you see just general immune activation. But interestingly, a lot of the specific pathways that are upregulated is what we already know from tuberculosis. And more interesting still is that when we compared the clarithromycin-exposed macrophages to those that had not been exposed to macrolides, there wasn't actually any change in gene expression at all. So that implicates, at least in this setting, they're not reacting to the macrolides at all. But this does show to us that upon encountering Mycobacterium avium, these macrophages do seem to respond as if it is something pathogenic that they need to deal with. So when looking at the bacterial response, we always first want to get an idea of just the general transcriptomic landscape between all the included conditions. So we made a principal component plot, which is shown on the slide. So the squares in the plot indicate the extracellular bacteria, while the circles indicate the intracellular bacteria. And in addition, the light blue samples are clarithromycin exposed, while the others were not. So the first thing that we saw was on the x-axis, which is principal component 1, which has about 50% of the variance in it. You see the effect in the infection itself, and you see that it's actually by way of, it's as big in the clarithromycin exposed as unexposed samples. While on the y-axis, you see the effect of clarithromycin, and it's quite clear that in the extracellular bacteria, the effect of clarithromycin is much more pronounced than on the intracellular bacteria. So I'll come back to that a little further on in my talk. So I'll first go in depth on the response of the infection. So moving forward, we determine the number of differentially expressed genes between each condition. And shown here on the right is the number of genes either upregulated or downregulated in intracellular bacteria in comparison to those grown in the absence of macrophages. And as you can see, a really large number of genes was differentially expressed. So around 340 genes were upregulated, while 250 were downregulated, which is about 10% of all the genes in the Mycobacterium avium genome. So again, we performed gene ontology enrichment on these gene sets, and we saw increased expression of antigenic proteins, including the PE, PPE proteins, increased expression of genes involved in nitrate reduction, and increased expression of the nitrate transporters NARC1 and 2. So for the sake of time, I'll only focus on what was upregulated in this talk. So first, the PE, PPE proteins and antigen expression. So PE, PPE proteins are actually proteins containing a proline glutamic acid or proline glutamic acid on their N-terminal. And they've been shown to play a really important role in recognition of tuberculosis, along with the other known antigens. So in intracellular Mycobacterium avium, we saw an upregulation of around 25 to 30% of all putative PE, PPE proteins and antigens, including the well-known PPE25 MAV protein that's actually previously already been shown to be contribute to the virulence of Mycobacterium avium. And the upregulation of these genes indicates to us, at least, that Mycobacterium avium is actively presenting itself to the macrophages, and it's not attempting to escape the immune response at all, as we do know many other pathogens do. And the second striking change in transcription we saw in the intracellular Mycobacterium avium was the increased expression of this nitrate reductase enzyme, known as a NARG to J, and the nitrate transporters NARC. So while it's well known that macrophages use nitric oxide as a defense, previous studies have also shown that that these high levels of nitric oxide can help control Mycobacterium tuberculosis infections by becoming cytotoxic. However, on the other side, it's also known that if they do fail to get high enough levels of nitric oxide, it can actually promote the survival of intracellular tuberculosis by helping to maintain redox homeostasis. And that's achieved as shown in the figure below. So the nitrate that's derived from this nitric oxide can be imported into the mycobacteria using the NARC transporters. It's then converted to nitrite using the nitrate reductase enzyme, and it thereby actually donates electrons to the electron transport chain and fuels ATP production. And the resulting nitrite can be transported back out again using the NARC transporters. This also shows that Mycobacterium avium is reacting to this high level of nitric oxide and is likely using upregulation of the system both as a protective measure for the nitric oxide itself, but also to adapt to the low oxygen tension that it often encounters within the macrophages. Again, quite similar to what we know from tuberculosis. And so lastly, I wanted to come back to the big difference we saw in the size of the effect of clarithromycin in intracellular and extracellular bacteria. So as you can see in the bar chart on the left, when we treated extracellular bacteria with clarithromycin, around 300 genes were up and down regulated respectively. But when you repeat the same analysis on intracellular bacteria, this is only about 20 and 10 genes respectively. And we believe this likely has to do with this high level of nitric oxide accounted within these macrophages and the effect that this has on the YB family of transcriptional regulators. So YB proteins are known transcriptional regulators of mycobacteria, and they regulate a wide scale of cellular processes, anything from cell division and metabolism to virulence and antibiotic resistance. So the YB proteins are relatively small and they contain 4 cysteine residues. And these bind to an iron-sulfur cluster, which is shown on the right side of the slide. So these iron-sulfur clusters are actually known to be nitric oxide sensitive, and exposure to this nitric oxide leads to conformational changes and then influences the transcription by these YB proteins. So the Mycobacterium avium genome contains 6 YB proteins, of which the precise function is not yet known. So we looked into our data and studied the expression of these proteins in 3 different conditions. So that of extracellular avium exposed to clarithromycin, of intracellular unexposed Mycobacterium avium, and intracellular Mycobacterium that has also been exposed to clarithromycins. And the heat map shows what we saw. So the log 2-fold change in expression of each YB coding gene is shown, where red is more highly expressed and blue is downregulated. And you can immediately see that the expression profile of the intracellular untreated and treated bacteria is much more similar than that of the 2 treated conditions. And we believe this is likely because the nitric oxide produced by the macrophages has changed the conformation of these YB proteins and is dominating further transcriptional regulation and thereby altering the normal antibiotic response. And this was of particular interest to us because that could actually mean that the effect of clarithromycin on Mycobacterium avium may be different intracellularly as compared to extracellularly, which is quite important to keep in mind in a lot of the in vitro experiments we do looking at susceptibility. So to conclude shortly, our findings indicate a few key responses that Mycobacterium avium has upon contact with host cells. So first, it seems to actively promote uptake, and then once intracellular, it utilizes the key host defense mechanisms to try and protect from the hypoxia encountered intracellularly. Interestingly, we didn't see any killing of Mycobacterium avium within our model, which is quite strange because when we could do the same model with other pathogens, you actually do see killing. So it just seems to be that Mycobacterium avium can resist this killing somehow. And the survival mechanisms we see actually seem to mirror what we already know from tuberculosis. The only exception is it's not actually able to outgrow the macrophages as tuberculosis is. So this really indicates to us that Mycobacterium avium is quite capable of surviving, it just lacks virulence. In addition to this, we showed that the intracellular environment does likely influence the effect of drugs and that we should keep this in mind when studying drugs in vitro. So moving forward, we would like to complete some experiments to look further into some of our findings. So we first plan on studying the generation of nitric oxide by Mycobacterium-infected macrophages over time. And secondly, we'd like to study the effect of this nitric oxide on the drug response in culture. So I just wanted to thank everyone who helped me in this project. So in particular, Valerie Kuchen, who helped me set up all the methods, and her supervisor, Einoud, from the Department of Internal Medicine, who all know everything about there is to know about the immune response. And from our department, Jordi Kohler, who is our bioinformatician, and of course, Jaco, my supervisor. So thanks for your attention.
Thank you very much. That's great lecture, really. And you both confirmed the right choice from the steering committee of the Congress. Really 2 great lectures. First, I have one question about NO, because it's quite interesting. There are some papers that suggest the use of NO as a, antibacterial factors to be used by inhalation, both in intensive care and non-intensive care, and even in cystic fibrosis. So what do you see in this area? Is there any possibility, or there is a substantial resistance by avium?
No, I think it possibly could help. It's hard to know exactly what the effect is, and I think that's important to look maybe more in humans, because this is obviously all in vitro, even though it does include human cells. I mean, in general, nitric oxide is cytotoxic, just in bacteria. Not all bacteria contain that, but mycobacteria do. They contain the nitrate reductase enzyme, so it is important to keep in mind that they can actually tolerate a certain level of nitric oxide, and it can be beneficial. So I think it could possibly be helpful, but you would need to have concentrations high enough that it would be more cytotoxic than beneficial to the bacteria.
You're muted, Francesco.
Yes.
Yes.
There's no question from the audience. Do you have any question?
I didn't see a question there, but I have a question and a comment.
No, I asked you if you have any. If not, I have another one.
Can I have one?
Sure.
Okay. Thank you, Jody. Congratulations for this very nice presentation as well. Congratulations for this award.
Thank you.
And I have a comment. Now, a question first and a comment second. This poor effect of clarithromycin that you have observed in intracellular Mycobacterium avium, can be extrapolated to other macrolides? This is the question. And the comment is, our industry in Boston is preparing a portable nitric oxide administration for COPD patients. I think that this could be, you should contact them, because this is important for the future therapies of these difficult to treat mycobacteria.
So sorry, your question was, if I understood correctly, what the effect of this might be on the macrolides in treatment, or?
No. In intracellular, because you showed that azithromycin is poorly effective in this, and if clarithromycin, these effects can be extrapolated to, for example, azithromycin, other macrolides.
Oh, sorry. Yes, I would assume so. Yes, because what we know from the macrolides is the bacteria respond to them quite similarly. It's hard to know because obviously now we're still looking at the transcriptome, so it's the reaction to the macrolides, and it doesn't say anything about the susceptibility to the macrolides, which is what we hope to look for in the future. So does it actually, the altered response to macrolides, does it actually alter the outcome of the treatment, which we hope to do soon in some in vitro experiments. Again, it's hard to translate that, I think, in patients, but in the lab, it'd be interesting to see what it means for susceptibility. But I would assume that azithromycin and clarithromycin both have a similar effect.
Okay, and the second, if you are aware of these portable devices for NO administration, because now NO is administered mainly in the intensive care unit, no, and in the anesthesia in the surgical room. But small industry in Boston is preparing that, well, has developed that for COPD. And this could be tested in Mycobacterium avium.
Yeah, I wasn't actually aware of that. So again, I'm not a clinician at all. I'm more a microbiologist, but it's definitely interesting to know. And I do think that obviously this is the preclinical, but it would be interesting to see what the effect of this nitric oxide is in avian patients, also because we know from TB that the effect.
Yes. I can pass the contact to you.
Yes, thank you.
And this forms part of the advice, no?
Yeah. Well, you receive a well done, Jody, from Jaco. That's good. And I have another question about, do you think that using other drugs in vitro, apart from macrolide, can you give any more information about the effect of this drug in blocking the activity of avium, or to improve the activity of macrophages?
Yeah, definitely. That would be very interesting. So the problem with this really is that the experimental setup, because you need enough cells from the donors, it's really hard to upscale this experiment. And we are looking into methods to be able to look at lower amounts of RNA using specific RNA probes, which would allow us to maybe include more conditions and go to much smaller sample sizes, because 10 million human cells is really like a huge amount. And so from one patient, we can only ever look at 4 conditions, which makes it hard to scale up to more drugs. But that's something we would definitely like to look at.
So I don't see any other questions.
No.
So I think we have to congratulate with our
a lot.
Really
Excellent
lecture. And I think this is a good start for our meeting. So we can start with the best. We started with the best possible lectures, and from 2 young ladies smiling. It is good. Even working with neutrophils and macrophages, you are still smiling. It is good.
Which is difficult, eh? Which is difficult.
Yeah. So I think we at the end of the day we can close the session. Tony, if you want to close, we can move to the.
No, thank you again for these excellent presentations. I think that this is the best way to start with this very interesting and probably applicable to the clinical practice in the future. So congratulations again, and an advice.
Thank you.
An advice, because we had to give an advice, Francesco. Says here in my, it's here, persistence and persistence in science. This is my advice.
Yeah, I buy it. Very good. Thank you very much. Thank you. Really.
Thank you.
Now we can move to the next section. I think we have a lecture now, so I can lead the. We have a question. Sorry.
Okay.
We have still a couple of minutes. Anthony De Souza, NTM are of course tricky to treat and are treated with multiple drugs. In practice, variable treatment regimens and adherence may mean incomplete dosing. What is the gene expression differences from Clari to say Clari plus sublethal aminoglycoside? Any clues about this?
No, so not yet. So at the moment, we have data on the response to pretty much all drugs used in treatment regimens. And moving forward, we want to look at, so in our center, we have a PK/PD model in which you can simulate drug treatment to all drugs using the concentrations you would find in the blood or in the lungs. And we would like to look at the transcriptome following treatment over time. So that's hopefully something for the coming year.
Good. Let me see if there is any other question. No.
Not really.
We can give the floor to Aurel Sibyl. Correct?
Yes, we are live now, I think. Yes, good morning or good afternoon, everybody. I'm Aurel Sibyl from Hospital Clinic in Barcelona, and it's my pleasure to introduce the next keynote lecture for Professor Tobias Welte. Probably all of you knows who is Professor Welte. He's the immediate past president of the European Respiratory Society, head of Department of Respiratory Medicine in the Hanover Medical School in Hanover, Germany, and among others, it's a member of the Hamburg Steering Committee and the member of the board of directors of the German Center of Lung Research. So it's a pleasure to introduce Professor Welter, who will talk about the new bronchiectasis management in the COVID-19 era. At the end of the session, we will have a few minutes for the questions, and you can send them by the platform. So, Professor Welter, go ahead.
So, thank you very much, Oriol, for the very nice introduction, and it's a real pleasure for me to be here and to give you a keynote lecture about bronchiectasis and COVID-19. As you all know, it's a very special situation. For 10 months now, we are in the corona crisis. We deal more or less every day with patients with COVID-19. It's a huge effort for everybody, for all the staff in the hospitals, for the physicians, the doctors, but it's also a challenge for the patients. And what I want to talk about during the next minutes is what will we do as a bronchiectasis patient with COVID-19. And this is a German slide. However, this is a publication we did for Germany some days ago about COVID-19 in patients with chronic respiratory diseases. And some of the issues I want to talk about will come out of this guideline paper. If you look what is known about bronchiectasis patients with COVID-19 in the literature now, it's not a lot. And therefore, I go back to the etiology of bronchiectasis. As you know, very different diseases, starting with cystic fibrosis, including asthma, COPD, immunodeficiency, tuberculosis, and other ones. And I will show you the data actually available for these kind of baseline diseases with regard to COVID-19. If you look, and all observational studies published go into the same direction, what the main risk factor for COVID-19 for a more severe course of the disease and for an increase in mortality is, it's not respiratory disease. It's on the first ranking places arterial hypertension and then metabolic disease like obesity and like diabetes mellitus. And there is a clear reason for this. If you look to the pathogenicity and immunology of COVID-19, it's an airway infection, no question about. But what is very special, very early in the course of the disease, it becomes systemic. Viremia is one of the key features of COVID-19. And when it is systemic, it affects no more the airway epithelial cells, but more the endothelial cells. And endothelial damage is one of the key factors of COVID-19. And this makes it clear why diseases which affect the endothelium, and this is true for hypertension and also true for metabolic diseases, are risk factors for a worse course of COVID-19. These are American data. These are our data from Germany published in Lancet Respiratory Medicine, also demonstrating that hypertension and diabetes is the key risk factor and chronic pulmonary disease is less frequent. Nevertheless, we have data for most of the baseline diseases which are important for the development of bronchiectasis. And I want to start with cystic fibrosis. 2 papers published looking to registries of cystic fibrosis patients. And in this one, you can see there is a very low number of cystic fibrosis patients affected by COVID-19, and the prevalence of COVID-19 in cystic fibrosis is lower than the prevalence in the population in general. And if you are a cystic fibrosis patient with COVID-19, your risk for severe course is very low. However, the 4 patients in this cohort which had been admitted to the intensive care unit all died. But this is not due to mainly COVID-19. It's something we know from cystic fibrosis, and I'm quite sure it's not different for Pseudomonas infected bronchiectasis patients, that these kind of chronic infection under mechanical ventilation is of risk, because there is a transmission from the bronchial bacteria from the Pseudomonas at a high bacterial load, a high bacterial density in the bronchi to the systemic circulation. And bacterial sepsis, and mainly Pseudomonas sepsis, is a very common finding in cystic fibrosis, but also in bronchiectasis patients who are ventilated. This is a second study from Spain out of a Spanish registry, and it's more or less the same. Only 8 CF patients with COVID-19 had been reported. The incidence is lower than in the general population, and none of these 8 patients had been admitted to the ICU and died. So in principle, bronchiectasis patients are not of increased risk for COVID-19 and worse course of COVID-19. One of the major, or 2 of the major baseline diseases for bronchiectasis are asthma and COPD. And asthma and COPD more or less are responsible for about 20% of all bronchiectasis patients. This had been one of the first papers looking to the adjusted risk ratio for hospitalization in the US with regard to different risk factors. And as you can see, again, hypertension, diabetes, obesity are number 1, and asthma and COPD does not figure out to be a major risk factor. And this is another paper looking to asthma alone, and as you can see, this is also not a significant risk factor for hospitalization for patients with COVID-19. In contrast, the risk for asthma patients for severe course of COVID-19 disease seems to be lower than the risk in the general population. And there had been a lot of controversies why this could be the case. And this is one of the main papers, a paper in the Blue Journal looking to the ACE2 receptor. The ACE2 receptor and its neighbor, the protein serine receptor 2, these are binding receptors for SARS-CoV-2. And in this work, it has been shown that ACE2 receptors are reduced in asthma patients, and this may be due to an influence of the Th2 response in asthma, which seems to be not correlated, associated with COVID-19 disease. COVID-19 disease, from an immunological point of view, is a Th1 disease, and in Th2 predominant patients, the likelihood for COVID-19 seemed to be less. This is the biggest study in obstructive lung disease from an UK cohort, an English cohort, looking to the influence of the baseline obstructive disease on COVID-19 disease, and especially here it is looked whether regular ICS treatment could be a risk factor for severe course of the disease. And this is the asthma pattern. And making a long story short, there is absolutely no risk of more severe disease and mortality in patients with asthma who are on inhaled steroids. And I think this is also important for the bronchiectasis community. If patients, for what reason ever, are on inhaled steroids, they should keep these medication. There is no increase in risk for COVID-19. And this had been confirmed in another analysis published here, demonstrating that in Italy, inhaled steroids does not increase the risk for COVID-19. I want to move forward to COPD, another analysis published 2 months ago in Journal of Allergy and Clinical Immunology. And again, what you can see here in comparison to patients without obstructive lung disease, in asthma, the mortality seems to be lower than in the control group. In COPD, it seems to be higher, but you have to have in mind that COPD patients are older and more comorbid than asthma and control patients, and mainly metabolic diseases and hypertension are very frequent comorbidities in COPD patients. And again, there's no influence in these patients when inhaled steroids had been used. Again, the Yaki analysis demonstrating that obesity is a risk factor, emphysema is not, bronchiectasis here shown for the only time not a risk factor for severe course of the disease, and the same is true for asthma and COPD, even in multivariant analysis. Well, one explanation why the COVID-19 pandemic and the recommendations and restrictions as a consequence of COVID-19 disease could be not bad, but good for bronchiectasis patients is shown here. These are data from Germany looking to GP visits for respiratory infections in Germany during different years. And what you can see is normally there is the influenza, the flu period from mid of January to end of April, and this had been the same during all of the years. This is 2020, and this is the 18th of March when in Germany the lockdown started. And what you can see is under the condition of a lockdown, and you had the same in most of the European countries, in Spain, in Italy, in France, in the UK, there is a drop down of respiratory infections to less than 20% of normal. And the same had been shown in Australia. These are the influenza patterns in Australia during summertime, which is winter time in Australia. This is a normal number of influenza patients, about 15 to 20,000 every year. And this is 2020, 187 influenza patients during the whole year here in Australia, and only 18 hospitalized. And this is a clear effect of the lockdown measures of mouth, nose mask, face masks, which drop down infections. What should we take from this for bronchiectasis? I think, which is an important message, is viral infection, even in bronchiectasis, and even bacterial colonization and infection plays a major role. Viral infection is a trigger of deterioration. And if you avoid viral infection, you avoid deterioration of bronchiectasis patients. And again, this is a study from Hong Kong, and we just submitted very similar data from Germany to the Lancet. This is the hospitalization rate, the gray curve with heart failure, and the black curve with exacerbation of COPD during the years. And this is 2020. And this is the date of lockdown. And what you can see is there is a drop down in the hospitalization rate for COPD patients by more than 80%. In Germany, it's dropped down to 8%, a drop down rate of 92% for patients hospital admitted with exacerbation of asthma and COPD. And this is a clear effect of face masking and lockdown strategy. COVID-19 and tuberculosis, as you know, in Asia, in low-income countries, tuberculosis is one of the main reasons for bronchiectasis. And this is a study from India published in the European Respiratory Journal demonstrating that there is COVID-19 in tuberculosis patients, and there is mortality due to this combination of tuberculosis and COVID-19. There is an increase in COVID-19 mortality in patients who suffer from active tuberculosis. This could be due to immunological reasons, due to structural damage of the lung. And the main message from this is, if you are an active tuberculosis patient and got COVID-19, the tuberculosis treatment must be remained. So you must stay with tuberculosis treatment, because a stop of the treatment may be very risky. Interstitial lung disease and COVID-19. This is a paper published in the Blue Journal some weeks ago, demonstrating that there is a risk for higher mortality in patients suffering from interstitial lung disease, but this is strongly related to age and to the impairment of the lung function before the COVID-19 disease. And again, if you look to the risk factors, patients who are obese, and obesity in interstitial lung disease is mainly due to long-term steroid treatment. So patients who are obese had a higher risk for mortality. And if you put this out, if you just for obesity, the risk is no more increased. What is with cancer patients? Is there any risk of cancer for mortality due to COVID-19? And the answer is yes. Cancer patients are at higher risk for COVID-19 and also for mortality, but this is related to the type of cancer. Hematological disease, leukemia, lymphoma have a higher risk than solid tumors, and it's related to the aggressivity of the treatment. If you are under aggressive chemotherapy with neutropenia as a consequence, your risk altogether is increased. But what is also shown is the likelihood to die from an aggressive tumor is higher than the risk of COVID-19, and therefore it's strongly recommended that patients, even with cancer, should be further treated with their standard regimen. These are the WHO guidelines for obstructive lung disease patients, and if I want to summarize bronchiectasis to this, it's the same recommendation. So Gold and the WHO both recommend to stay with the common treatment, to stay with bronchodilators, with inhaled antibiotics, with everything else. Maintain your therapy and do not stop it due to COVID-19. And I think that's also true for all of our bronchiectasis patients. So recommended treatment should be continued, and this includes nebulized treatment. Yes, nebulizers produce aerosols, and aerosols are of risk to spread infectious particles. However, if you keep your hygiene measures, your prevention recommendations, then you could use aerosolized treatment as normal. With this, I want to move to my last point, and this is long COVID or post-COVID syndrome. And this is something which may be of major interest for us, for the bronchiectasis community. If you look to the data published for the post-COVID period, this is one of the first papers from Italy, and it's very threatening because they stated that more than 80% of patients suffer from ongoing symptoms weeks after COVID disease. However, in this study, only patients who were hospitalized and most of the patients coming from ICU had been studied. In another study coming from Egypt, it's a little bit a different picture. In this study, it's shown mostly in outpatients that there are ongoing symptoms, but the most important ones are fatigue, anxiety, and some very unspecific syndromes. Well, fatigue is a very difficult symptom, and it's well known from other infectious diseases. I have seen fatigue patients after mononucleosis, after CMV infection, after other viral infections. However, it's very often seen in patients after COVID-19 disease. And we use in my department a fatigue assessment scale to quantify this. This is a scale which comes from sarcoidosis, where also fatigue syndromes are very common and is now used in COVID-19. Altogether, I would say about 1 to 2% of all COVID-19 patients suffer from fatigue long-term after the COVID-19 disease. And what's very interesting is this fatigue is not associated with the severity of the COVID disease. It also occurs in patients with mild disease. We use a very specific rehabilitation program focused on exercise training, on muscular training, also respiratory muscle training, and on cognitive, neurocognitive functions to improve these patients, but nothing is established. However, I want to move forward to structural damage after COVID-19, and this is a paper again published in the European Respiratory Journal looking to lung function impairment after COVID-19. And what you can see here, the most important one is the impairment in diffusion capacity, and it's more impairment in patients who had been more severely ill, but there's no control group. My personal feeling as someone working in ICU for a very long time is that everybody who is mechanically ventilated for 2 or 3 weeks with a long time of recovery has such kind of diffusion capacity impairment. However, this is the main finding, and the pathological explanation for this is shown here. This is from a Chinese paper demonstrating that this is typical COVID-19, and then over weeks there are some residua, and then the chest CT is cleared up. However, in some patients, it's not totally resolution. There are pathological repair present. And this has been shown in a case report published here. And interesting for us, the bronchiectasis community, is that bronchiectasis, very nicely shown here, is a feature of pathological repair and repair defect in patients after COVID-19. In a recently published review, in expert review of respiratory medicine, all the data had been put together, and this is the conclusion out of this review. A very small number of COVID-19 patients, but what does small mean, having in mind that we have now 100 million of COVID-19 patients worldwide? In a small number of patients, there is residua after COVID-19, and these residua also include bronchiectasis, which is in line with what we see in post-infective bronchiectasis for other pulmonary infectious diseases. If this is the case, and with the high number of COVID-19 patients in mind, we have to be very cautious in the future to identify these patients early and treat them according to the recommendation. And here is a nice recommendation from the British Thoracic Society how to deal with patients after COVID-19. And this is the same we do in Germany. So if the patients are still symptomatic after 4 or 6 weeks after COVID, we perform a lung function test. If the lung function test is pathologic, if it is a slight impairment, mainly of diffusion capacity, we start a control 6 weeks later. If it is severe impairment, or if after 12 weeks there's still an impairment in lung function parameters, we perform a chest CT, and then the patient is treated according to the chest CT findings. And in patients in which nothing could be found in the lung, you have to consider that there is an involvement of the myocardium of COVID-19 and have to start a cardiological diagnostic. So, to finalize, bronchiectasis patients are neither at risk for COVID-19 nor for more severe course of COVID-19. This is the good message. Maybe they are in a way prevented by being more careful in patient contacts, so better adherence to the lockdown measures than in others. And what I said, the underlying environmental immunological milieu in bronchiectasis may be protective by suppressing Th1 lymphocytes. In general, treatment for bronchiectasis should be continued as recommended. There's no reason to stop any of the treatments which we normally use for COVID-19. Even in patients who are SARS-CoV-2 positive, we should maintain the bronchiectasis treatment. Bronchiectasis could be one of the pathological features after COVID-19 infection. And this could increase the number of bronchiectasis patients worldwide during the next years. This is something we have to be aware. And if typical symptoms we all know, sputum, dyspnea, high sputum volume, hemoptysis, and the others, if this occurs, we should start a diagnostic procedure, including chest CT, for these kind of patients. COVID-19 is a total new disease. SARS-CoV-2, a totally new virus. We learn every day something new about this virus. And because of this, the very quickly increasing knowledge of COVID-19, the news of yesterday are old, and everything could be different today and will be different in a week or a month. So we have to adapt our recommendations in concordance to the knowledge coming up day for day. I want to thank you very much for your attendance. I thank the organizers, my dear friends Francesco Blasi and Tony Torres, for setting this up in very difficult times. Thank you to Oriol to sharing me, and I'm convinced there will be a post-COVID time and we will meet again, and I'm today look forward to the next meeting face-to-face with all of my friends. Thank you very much for your attention. You are muted, Oriol.
Yes, now, thank you very much, Tobias. Sorry. Thank you very much for this wonderful and clear presentation about these two topics that all of us are very, very involved and interested. I don't know if there are questions from the audience. We have a very few minutes. Let me see. Yes, my question is a very clinical question. Of course, there is no evidence and only recommendations, but in which patients we should suspect bronchiectasis after the COVID-19. We have to guide by the symptoms and the lung function test, and at what moments should we perform a CT scan looking for bronchiectasis, what we should do in the clinical practice.
Yeah. Well, I think there are two major items. One, again, is diffusion capacity. We know from mainly Stefano Aliberti's work that the impairment in diffusion capacity is also something which is very special for bronchiectasis. And very often there's more diffusion capacity impairment than an obstructive lung function impairment. So we should perform lung function and we should do body plethysmography to get all the data. The second, and I think that's really important, sputum production is not typical for COVID-19. And if after the COVID-19 disease, there is a remarkable amount of sputum in these patients, you have to have bronchiectasis in mind, because this is not normal for COVID-19. Normal is a dry cough and not a lot of sputum. And if sputum appears for the first time and it's a high sputum volume, you have to have bronchiectasis in mind, and a CT scan in my mind is necessary.
Okay, thank you very much. We are on time and we have to move to the next presentation. So thank you all, and let's move on.
Keep safe, and seeing you tomorrow for the next lectures. Bye-bye.
Hello, everybody. Welcome to an exciting afternoon session on novel diagnostics and assessment methods. My name is Felix Ringshausen. I'm, as my chief, Tobias Welte from Hanover in Germany as well. And I'm on the one side, as already mentioned by the others, sad that we cannot meet in person this year and exchange as we were used to in the past years. But on the other side, I'm proud to be part of this global community of physicians and researchers caring for this neglected disease, even in difficult times like these. And I would like to introduce my co-chair, Michael Lobinger from London. So Michael, up to you to introduce the first speaker, please.
Thanks very much, Felix, and I echo many of those views too. It'd be much better to actually be somewhere nice and sunny seeing you all. So look, I'm delighted to introduce the first speaker. Now, actually, the first speaker is a recorded presentation, but I've been told that for questions at the end, this speaker will be available. The title of his talk is Lung Function Testing Findings, and it's going to be given by Professor Y.G. Guan, who's an associate professor and principal investigator of the first affiliated hospital of Guangzhou in China. So hopefully that will just start running.
Hello everybody, I'm Dr. Weijie Guan from the State Key Laboratory of Respiratory Disease, the first affiliated hospital of Guangzhou Medical University. So first of all, thank you very much for inviting me to participate in the World Bronchiectasis Forum. So it's a great opportunity for me here. And today's topic is all about lung function impairment in bronchiectasis. So first I would like to declare that I will receive the following grants. However, I have no other potential conflict of interest associated with my topic. As to the best of knowledge, bronchiectasis is no longer regarded as an orphan lung disease, but alternatively, it's really common disease, largely neglected by the respiratory clinicians. So according to the vicious cycle, due to the impact of the primary or either the secondary etiologies, so a perpetual cycle prevails in patients with bronchiectasis, which results in permanent airway inflammation, bacterial or infection, as well as the airway destruction. Such a relentless cycle would ultimately and inevitably result in lung function impairment. For instance, a study by James and colleagues showed that higher neutrophil elastase activity correlated negatively with lung function parameter, the FEV1. And more importantly, it has already been shown that the greater FEV1 decline in patients with bronchiectasis who had a high level of baseline neutrophil elastase activity are strongly correlated with each other. And more importantly, the colleagues from Spain, for example, Martinez Garcia and colleagues have also shown that there have already some of the key clinical factors associated with faster annual FEV1 decline in patients with bronchiectasis. Four most salient factors associated with more rapid FEV1 decline consist of either the Pseudomonas aeruginosa colonization, more frequent previous exacerbation frequencies, higher baseline FEV1 level, and finally, more prominent systemic inflammation. Nevertheless, Pseudomonas aeruginosa colonization always remains a significant risk factor contributing to more rapid lung function decline. And more importantly, when we take a closer look at the two prevailing severity rating scores, including the BSI developed by James and colleagues, and FASET score developed by Martinez Garcia and colleagues. So FEV1 constitutes the central and overlap element for both the disease severity metric assessment. So these addresses the importance of lung function assessment. So overall lung function is a very gross term that consists of various domains. For example, outlines here, today I would like to briefly introduce to you some of the key findings related to different domains of lung function impairment in patients with bronchiectasis. Now first take a look at the ventilation and diffusing capacity. According to one of our previous clinical study, it has already been shown that even when remaining clinically stable, a number of bronchiectasis patients will have lung function impairment. These constitute either the restrictive or obstructive or even mixed patterns. For example, in patients with bronchiectasis, approximately 15% of patients will have restrictive disorder, and slightly greater than 20% of patients will have obstructive disorder, and slightly less than 30% will have mixed patterns. Nevertheless, factors associated with severe F1 decline consist of either the prolonged symptoms or either Pseudomonas aeruginosa colonization or a greater HRCT score. It should also be addressed that a number of patients will also have impaired diffusing capacity even when remaining clinically stable. And the factors associated with a decreased diffusing capacity only consist of a great number of bronchiectatic loads, suggesting the magnitude of airway destruction correlated significantly with the magnitude of lung function decline in patients with bronchiectasis. Having demonstrated the magnitude of disorder associated with ventilation diffusion, we next go on with ventilatory heterogeneity, which can typically be measured via either the chest HRCT assessment, which can be difficult for quantification, a residual volume measurement, which may also be time-consuming, the single breath measurements, for example, phase 3 slope, which has less been applied in clinical practice, and finally, the lung clearance index, which involves multiple breath washout tests. Now here, I would like to show you the clinical importance of measurement with lung clearance index. So this is a technical jargon, which essentially it should be acknowledged that this should be a very complex measurement. But importantly, it should be addressed that the LCI, lung clearance index, is a highly repeatable measurement and is a metric that correlates significantly and negatively with F1. LCI, more importantly, it more sensitively identifies either mild or early stage lung function impairment than our gold standard FU1. So some of the scholars have also proposed the use of LCI as an important endpoint for a clinical trial assessing the efficacy of pharmaceutical intervention in patients with bronchiectasis after either the chest physiotherapy or other pharmaceutical interventions. So regardless to say that there are other papers that have shown the superiority of assessment with the LCI as compared with our F1, the gold standard. Another study demonstrated the different magnitude of changes in LCI in patients with bronchiectasis, cystic fibrosis, and those of healthy controls. And concurrently, the FU1 correlated negatively with LCI. And finally, it's interesting to know that the magnitude of disorder associated with LCI was highly comparable between patients colonized with other potentially pathogenic microorganisms and those without, suggesting that the bacterial spectrum might not correlate significantly with the LCI. And furthermore, a study by Grillo and colleagues showed further confirmed the high repeatability of the LCI measurement. And they also show that the LCI correlates significantly with other various lung function parameters. But importantly, the study failed to demonstrate that the LCI can improve significantly after the chest physiotherapy for the management of exacerbation of bronchiectasis, with the detailed mechanism underlying the observation remaining largely unclear. And finally, our previous study have shown the comparability between the LCI measurement and the MEF in the resource-restricted areas. So we have proposed that in the resource-restricted areas, MEF might act as a surrogate of LCI for the clinical assessment of magnitudes of ventilatory heterogeneity in patients with bronchiectasis. And more importantly, neither of the present study nor the previous research has confirmed the role of LCI in evaluating the acute exacerbation or convalescence after therapy. And furthermore, because conventional measurements only rely on the measurement of either ventilation or diffusion, they have neglected largely the airway resistance. But to the best of knowledge, some of the clinical assessments, for example, the impulse oscillometry, has already been made available. Impulse oscillometry, or IOS for short, could differentiate the characteristics of airway resistance and can clarify the most likely location of increase in airway resistance. So based on this research hypothesis, we have conducted a cohort study which demonstrates that IOS could really differentiate patients with bronchiectasis from those of healthy controls. And more importantly, we reaffirm that the IOS parameters, except for the airway reactance, correlated strongly with the disease duration, HRCT score, BSI, and the FEV1. And more importantly, we found that the IOS score more sensitively identified early stage or either mild bronchiectasis as compared with our gold standard FEV1. And finally, we failed to confirm that the IOS parameter changed dramatically during bronchiectasis exacerbation. More importantly, during clinical practice, we may also readily notice that some of the patients might also have concomitant airway hyperresponsiveness. But to the best of the knowledge, such study findings are really scarce. There has only a handful of research information available. According to the research by Balkan and colleagues, patients who have airway hyperresponsiveness would demonstrate a greater magnitude of impairment of FEV1 at baseline and poor quality of life. But how these findings translate into clinical practice remains largely unclear. So further research is definitely warranted. So in case some of the patients will have notable airway hyperresponsiveness, it would not be surprising to anticipate that a subgroup of patients will also have notable bronchodilator response. So in fact, our previous study has demonstrated that approximately 14% of patients, even when remaining clinically stable, who have undergone bronchodilation tests will demonstrate significant bronchodilator response. And importantly, we have shown that the magnitude of the bronchodilator response did not correlate significantly with the sputum or blood eosinophil count. And interestingly, we did not find any notable difference in FEV1 decline between patients with or without significant bronchodilator response. But contradictorily, we noticed a lower instead of a higher exacerbation risk in patients with significant bronchodilator response, with a detailed explanation being still largely unclear. Anyway, some of the previous research also seemed to be in favor of our finding. For example, a study by Hassan and colleagues also suggested a number of bronchiectasis patients will have also concomitant airway allergies or atopy. And these patients might readily demonstrate significant bronchodilator response. And the author suggested bronchiectasis patients should routinely undergo bronchodilator testing should airflow limitation be indicated through spirometry testing. And furthermore, the colleagues from Korea also suggest an even greater subgroup of patients would have a great response to bronchodilators. And authors also indicate that the magnitude of a bronchodilator response to, for example, salbutamol might herald the therapeutic response to LABA or LAMA in the long term. So whether these findings can translate into our routine clinical practice remains unclear because the study is still limited by this design. For example, it's relatively short duration of observation and relatively small sample size. And finally, we have also recently demonstrated that in bronchiectasis patients who have hypocapnia throughout the exercise testing, for example, with either the treadmill exercise or cycle ergometer, these patients would have marked exercise intolerance, even despite the lack of overt right heart failure. So these suggest the potential role of the cardiovascular pulmonary exercise testing in bronchiectasis patients who have no overt signs of heart failure. Whether the clinically a subclinical heart failure would precede the development of other clinical symptoms in these patients would be likely involved in further studies. Because the CPET, the cardiopulmonary exercise testing, would be sometimes complicated, which we usually require the dedicated personnel or either the intricate facilities inside dedicated medical teams. So usually the simpler tests, for example, 6-minute walk tests, may be preferred in the resource-constrained regions. So we have also previously demonstrated that the 6-minute walk test in patients with bronchiectasis did correlate significantly with the magnitude of impairment in spirometry and the quality of life as assessed with St. George's Respiratory Questionnaire total score. And we have also revealed that the two factors associated with a decreased 6-minute walk test, walk distance in patients with bronchiectasis mainly consists of either impaired diffusing capacity or a dyspnea at baseline. However, it is interesting to know that for patients who have an equivalent FEV1, patients with bronchiectasis will inevitably have the longer 6-minute walk distances compared with patients with COPD. However, the precise mechanism, especially pathophysiology mechanism, underlying our observation remain largely unclear. And finally, the colleagues from Italy have also summarized a key finding, and the paper has already been published in Respiratory Medicine. The central idea of the paper showed that the pathophysiological abnormalities in adults with bronchiectasis were pretty often overlapped instead of isolated finding. So the authors have also reviewed greater magnitude of reversibility for patients with an RV assessment rather than the FEV1 assessment. So this addresses the potential role for adding further assessment, including the slow lung volume assessment over spirometry in patients with bronchiectasis. And more interestingly, there should be patients would have a more severe airway obstruction and air trapping in patients with reversibility in residual volume instead of FEV1. So the key idea is that lung function abnormality usually overlaps in patients with bronchiectasis. And the fact that this has been largely neglected might have likely stemmed from our ignorance of the critical importance of lung function assessment in patients with bronchiectasis. However, this finding does not negate a one-size-fits-all solution that indicates that all items of lung function should be tested in bronchiectasis patients. Rather, I would personally recommend a personal medicine approach, the so-called targeted assessment based on the science. So this might represent a better approach for diving deeper into the pathophysiological abnormality in patients with bronchiectasis. And finally, an interesting finding, as have addressed previously, was the changes in lung function during exacerbation. Many of us might have been largely confounded by the difference in findings as compared with other chronic airway inflammatory diseases such as asthma and COPD. So to the best of knowledge, patients who have asthma and COPD would typically demonstrate an abrupt change in lung function parameter during exacerbation, which progressively recovers during convalescence. Nonetheless, this is totally never ever the case in patients with bronchiectasis. And these findings also extend starting from ventilation to the ventilatory heterogeneity, airway resistance, and even including small airway parameters. None of these parameters show both clinically and statistically significant changes and exacerbation and during convalescence. But nonetheless, detailed pathophysiological mechanisms underlying all our observation are unknown. And of course, we are not in a very position to explain the detailed mechanisms of these findings. And despite all these findings, should there be further room for identifying positive signals? We say, yes, things might be. Because the study by Brill and colleagues showed an abrupt decrease in PEF, the peak expiratory flow during exacerbation in adults with bronchiectasis. But, well, to the best of our knowledge, the PEF assessment has typically been applied in patients with asthma and COPD. So exactly, very few of us would apply similarly to patients with bronchiectasis. So perhaps this would offer us at least some of the positive signals for our future clinical assessment. So there remain many of the open research questions that has already been delineated on the slide here. So I will not read them out individually. But to sum up, we have shown that lung function impairment is really common and pretty often overlap in patients with bronchiectasis. So many lung function parameters correlate significantly with bronchiectasis. And there has been a limited reduction in lung function parameter at the onset of exacerbation and including the convalescence, which is not shown in the slide here. And finally, any ROS parameter being abnormal correlates more sensitively identifies the early stage or mild bronchiectasis. So that offers some rooms for other measurements. Meanwhile, we should also pay attention to the bronchodilator response and the impaired exercise capacity. So these all represent many unmet clinical needs that may warrant further investigation. So I would like to extend my thanks to all of our colleagues here and those who have been working along with our research group. So thank you once again for your participation. We look forward to meeting with each other.
Okay, I hope we are live. I thank Professor Guan for providing this pre-recorded excellent talk. And I would like to remind you, you have the chance to ask questions in the chat. And in the later course of this session, Professor Guan will join us and will be able to address these questions. It's an honor to introduce the next speaker, a man who actually needs no introduction, and I'll do my best to introduce Professor Stefano Aliberti from Milan, one of the most productive heads in bronchiectasis and respiratory infection research, and one of the driving forces in clinical care in Europe and globally. To say he has been head of the Respiratory Infections Assembly of the ERS in the past and keeps himself busy with multiple positions. I'm always absolutely wondering about your productivity, Stefano. He will be addressing the question of biomarkers to assess the severity and the prognosis of disease. The floor is yours, Stefano. Thank you.
Thank you so much, Felix. I don't deserve this really nice presentation. Thank you so much. Thank you, Michael. Thank you, Professor Blasi and Professor Torres for putting this virtual congress together. And let me thank also Public Reaction, Marcia, and all the team for the great job they did also for this virtual meeting. You guys are terrific. Thank you so much for what you're doing every day. So I will try to explore with you a couple of ideas on the use of biomarkers to assess severity and prognosis in bronchiectasis. These are my conflict of interest, and I think this is the most important slide in my presentation. And actually, this slide put together the long path, the long way from the analytical validation to the clinical validation and back of a biomarker in medicine in general. Let me tell you this. I think that there are plenty of biomarkers that we know in bronchiectasis, but the majority of them are in the early phase. Most of them are coming from single centers experience with no validation cohort, without any kind of data in terms of implementation in clinical practice, with measurements that have been done across different matrices. And I will try to address some of these biomarkers in my presentation, moving from the most solid one to the newest one. When we evaluate biomarker, just to remind you, we need to be sure that the biomarker is able to make to define a difference between the disease and the healthy subject. Is important because we need to reproduce the biomarkers across different settings, and the biomarker at the end is important not only to predict disease severity, not only to predict clinical outcomes, but qualified biomarkers is the one that is used to develop a drug and to control the implementation of a specific drug. Most of the biomarkers we have in bronchiectasis are mainly are touching mainly the inflammatory picture of the disease. And coming from also the story of other chronic respiratory diseases like cystic fibrosis, neutrophilic inflammation has been the most touched area where biomarkers have been developed. And I will be focused my presentation mostly on proteases, including neutrophil elastase, cathepsin G. I will touch some nectosis, and I will also try to explore not only sputum as a matrix, but also saliva and blood. So as I mentioned, neutrophil elastase has been the most studied biomarker in bronchiectasis so far, and has been shown to be associated with disease severity. These are the first data came out 3 years ago in the Blue Journal, where the James Chalmers group published a paper showing a correlation between the activity of neutrophil elastase in sputum and disease severity measured through the BSI. This is the left part of my slide, where you can see the correlation, the median level of neutrophil elastase across the mild, moderate, and severe BSI groups. And for the first time, this data have been validated also across different cohorts, and specifically from Milan in Italy and from Barcelona in Spain, with this paper published recently by Andrea Gramegna. You can see on the right part of the slide, the bronchiectasis patients have been divided in 3 groups: those with low, medium, or high and sputum neutrophil elastase level. And you can see that not only the bronchiectasis severity index, but also the E-phase were statistically different across the 3 study groups, with an increase of people with in the severe group, BSI and phase, moving from the low to the medium and to the high neutrophil elastase group. So the BSI and the phase are multi-parameters kind of scores, and one of the components is the radiological involvement. So after this experience, we decided to look specifically in details on the relationship between neutrophil elastase and radiological severity. So we review 165 CT scans from adults bronchiectasis patients in the Milan and Barcelona programs. All these CT scans have been reviewed by 2 radiologists from Milan and 2 radiologists from Parma, who scored for BALA, the modified RAFE, and the BRICS score all the CT scan. And we were able to correlate this with the sputum levels of neutrophil elastase. And as you can see from the table, there was a significant correlation between the activity of neutrophil elastase in sputum and the BRICS, the BALA, and the modified RAFE. The box plot at the bottom of the slide are showing the level of neutrophil elastase in patients who have been classified according to the BALA on the left, the modified RAFE in the median, and BRICS on the right, with mild, moderate, or severe impairment. As you can see, that there was an increase of a medium level of neutrophil elastase across the 3 study groups and across the three different scores. So this is important to me because in some patients we are not able to get sputum very easily. And the CT scan actually is a very important tool we have in clinical practice. And if these data will be published and will be validated in different and external cohorts, then we will be able to define a threshold of disease severity in terms of radiological appearance that will maybe select a patient with a high neutrophilic inflammation who might deserve a special treatment. The neutrophil elastase in the sputum is associated with a specific microbiome pattern. These are the data by Martino Oriano published this year in the European Respiratory Journal. This was an observational cross-sectional study conducted at the bronchiectasis program in Milan, and neutrophil elastase was evaluated in sputum of bronchiectasis patients during stable state. And in the same sample, the microbiota was analyzed through the 16S RNA gene sequencing, but also we evaluated the molecular assessment for different respiratory bacteria through real-time PCR, along with a series of different clinical data. So the figures at the right part of the slides are showing that a decreasing alpha diversity, in this case evaluated through the Shannon index, but also the PLO evenness or the richness gave the same results, was significantly correlated with an increase in elastase. And then we also found a distinct microbiome pattern within the 2 study groups, those with high and those with low neutrophil elastase. This microbiome pattern was mainly characterized by an enrichment with Pseudomonas in the high elastase group and with Streptococcus in the low elastase group. And then we confirmed this data with real-time PCR, showing an association of Pseudomonas aeruginosa with elevated active neutrophil elastase levels, based not only on real-time PCR, but also on standard culture. The neutrophil elastase also correlates with clinical outcomes. These are well-known data published by the same group, published in the Blue Journal in 2017, showing an association between neutrophil elastase and clinical outcomes in terms of time to first exacerbation, hospitalization, and also all-cause mortality. And these data are more important if we think that we can measure neutrophil elastase in our clinics. This is the NETSTIC, the neutrophil elastase airway test, with the different grade from 0 to 10, data published by Amelia Schoemark in the ERJ last year, showing showing different things. First, there was a correlation between the neutrophil elastase activity measured through the NETSTIC and the disease severity evaluated through the BSI. And then the time to first exacerbation has been reported in the Kaplan-Meier according to the baseline NETSTIC neutrophil elastase results. And as you can see, those with a positive more than 0 or high more than 6 of neutrophil elastase results, and the NITSTIC had a worse outcomes. And neutrophil elastase is also important because now we might have promising data on the use of brensocatib in patients with bronchiectasis. So it's very important to give the right drug to the right patients, and we have a tool that can stratify patients according to neutrophilic inflammation. Another important biomarker in the serine protease group is cathepsin G. These are the data coming from a multi-center observational cross-sectional study. 320 patients have been evaluated for cathepsin G in sputum during stable state. We found that 77% of the patient had detectable levels of cat G in sputum with a median levels of 75. And we found different things. So the different figures show you a correlation between cathepsin G and disease severity with the BSI, cathepsin G with quality of life, and then you can also see that we explore the interpatients diversity through the PCOA analysis based on the Bray-Curtis dissimilarity, and we show that there was a clustering between samples with similar levels of CAT-G through the Permanov analysis. The bar plots, the abundance are clearly different between patients with low, medium, and high cathepsin G levels. And also we took a look at the clinical outcomes during a follow-up. These are the data from Dundee showing that there was a decreased time to first exacerbation among patients with high level of CAT-G in sputum. Interestingly, we also compare the performance of these two biomarkers, neutrophil elastase and cathepsin G, showing actually no big differences in predicting disease severity, chronic bacterial infection, hospitalization, and also radiological involvement. Another important area that had been studied, especially from the Spanish colleagues Oriol Sibila and Lidia Perea, is the role of antimicrobial peptides. We know that the antimicrobial peptides are linked with disease severity and phenotypes in bronchiectasis. The paper published by Oriol Sibila in Thorax a few months ago, but these that I'm showing you are brand new data that Lydia put together and published in CHEST a few weeks ago, coming from a prospective multi-center cohort study where they asked themselves if they were able to identify clusters of patients based on levels of antimicrobial peptide, but also airway inflammation, tissue remodeling, and damage. And they found 3 different clusters after a 2-step cluster strategy. And they show that these 3 clusters of patients can stratify the population, are associated with disease severity, and are also associated with exacerbations. Holly, Dr. Kair, a few minutes ago during the Rising Star session, talked to you about the endotyping bronchiectasis through a multi-omic profiling, and let me congratulate with Holly for this incredible job and great presentation she did. Moving from sputum to serum. I'm sorry, this is a typo. This is not sputum, but it's serum. Desmosine has been one of the most interesting biomarkers evaluated, showing an association with disease severity, but also an association with long-term all-cause and cardiovascular mortality. These are the data coming from the Taybridge bronchiectasis registry on the left, and these are the data that Dr. Huang put together in a research letter published in the Blue Journal this year, showing that increased levels of desmosine were associated with an increasing all-cause mortality, and this relationship was also persistent after adjustment for baseline disease severity. So very, very interesting data. In serum, we also have blood eosinophil. We have some idea of blood eosinophils in COPD and asthma. These are one of the few data looking at this biomarker in predicting an intervention, which is inhaled fluticasone response in bronchiectasis. We have Miguel Angel Martinez Garcia. We put together an unplanned post-hoc analysis of a randomized control trial, which looked at the inhaled fluticasone intervention for 6 months, having as a primary endpoint a significant change of the St. George Respiratory Questionnaire. And then we stratified people with low eosinophil, less than 3%, versus high eosinophil, more than 3%. And we found that the primary endpoint, the changing in St. George, was only statistically significant among those with a high eosinophil at baseline. Always in the blood, we can also measure platelets. We know that thrombocytosis, not only in chronic respiratory disease like in COPD, but also in acute disease like pneumonia, has a special role. And we demonstrate in more than 1,700 bronchiectasis odals across 10 centers in Europe that thrombocytosis is associated with hospitalization, but also with both 3-year and 5-year mortality. So thrombocytosis as a immunomodulatory, anti-infective kind of biomarker. And finally, we move from serum, from blood, also to saliva. We already talked about the airway antimicrobial peptide, and this is the data that Lydia Pereira recently presented at the US Congress. They evaluate across 157 adults with bronchiectasis salivary, the salivary sputum secretory leukocyte protease inhibitor, and LL37, which are antimicrobial peptides. And they found that they are their level in saliva are correlated with the bronchiectasis severity index, but also patients with chronic Pseudomonas aeruginosa infection also at the lowest level of salivary SLPI. And they also found a weak but statistically significant correlation between salivary and sputum SLPI and LL37 between sputum and saliva. So these are interesting data. So to conclude, I hope that I give you an overview on the current data on biomarkers in bronchiectasis. And most of these data are coming from a network of people who are working together under the head of the European Respiratory Society and the BRIDGE study, what is the EMBARK 2 CRC. A lot of data are also will also come in the next future, thanks to the integration of the different omics. I didn't have the time to talk about cilia, the cilia sub-study, and also more in detail about the microbiome as a biomarker in bronchiectasis, but I think that you will have in the next future time to see the results from the EMBARK BRIDGE study. And finally, let me thank all the bronchiectasis team in Milan, Professor Blasi, Dr. Oriano, and Giovanni and Soggio and Laura Saderi, but also the friends we are working together, we are working with in Barcelona, in Dundee, in Singapore, and in New York, all the people and all the friends from the EMBARK and the European Respiratory Society. And thank you again for your attention.
Thanks very much, Stefano. I'm live now. Thanks very much indeed, Stefano, for an excellent talk, very comprehensive, and I'm sure that will generate a load of questions for the end when we can discuss all the different markers and measurements together. Moving on now away from samples and more into radiology, we have a talk by Dr. Meerberg now. Dr. Meerberg is a PhD student and medical doctor working in the research group of Harm Tiddens in the Erasmus Medical Center in Rotterdam. And she's going to be talking about quantitative chest CT scoring technique for bronchiectasis. Jennifer.
Yes. Okay, can you now see my screen? Okay, I think I'm... Okay, good afternoon, everyone. I'm Jennifer Meerberg. As Professor Lubinger told, I'm from the Erasmus Medical Center in Rotterdam in the Netherlands. I would like to thank you very much for giving me the opportunity to present in this session. And today I will show you how we developed a quantitative chest computed tomography scoring technique for bronchiectasis, the best CT method. First, I will start with some background information. As you all know, bronchiectasis can be caused by a wide range of underlying diseases, and this results in a heterogeneous patient population. We know that Pseudomonas aeruginosa is a common pathogen in BA patients and is associated with quality of life and with a reduced quality of life and shortened life expectancy. However, to date, there are no registered therapies available for bronchiectasis patients with a PA infection. And an important reason that there is a lack of sensitive outcome measurements, specifically sensitive imaging outcome measures that can be used in clinical studies. The gold standard to detect BE is chest computed tomography. And as bronchiectasis is a radiological diagnosis, chest CT scans contain valuable information that could be used to phenotype BE patients, and furthermore as outcome trials and as outcome measure in clinical trials. So the aim of our study was to develop a sensitive CT outcome measure to phenotype and to quantify structural lung disease in BE patients. And this resulted in the bronchiectasis scoring technique for CT, in short, the best CT methods. So our study was a sub-study of the IBEST. The IBEST study was there to evaluate the efficacy, safety, and tolerability of tobramycin inhalation powder in BA patients with a chronic BA infection. The study population consisted of patients with an age above 18 years with a proven diagnosis of bronchiectasis confirmed on CT scan and with a proven chronic BA infection. Main exclusion criteria were a diagnosis of cystic fibrosis, asthma, or COPD with a smoking history of at least 20 pack years. So for the best CT method, we collected the most recent CT scans of patients enrolled in the IBEST study. Inclusion criteria were a digital format, a volumetric protocol, and a slice thickness of at least or less than 3 millimeters, not more. And CT scans with severe motion artifacts or an incomplete display of the lung were excluded. Both inspiratory and expiratory CT scans were analyzed. CT scans were analyzed with several scoring methods, which will be explained in the following slides. First of all, obviously, we analyzed the CT scans with the newly developed best CT method. Furthermore, we used the Hartmann method. We measured the intra-branch tapering, and furthermore, to assess the reproducibility of the best CT method, selection of 20 scans were rescored by the same observer and by a third observer. We then compared the best CT scores with the following clinical data. The FEV1 percentage predicted in GLI units, the bronchiectasis severity index, the PA bacterial load in colony forming units, the quality of life using the quality of life questionnaire from bronchiectasis from Alexander Quidner, and furthermore the exacerbations during the study. Well, this slide shows the best CT method. The best CT is a grid-based method, and as is shown on the figure on the lower right, a grid is placed on the actual CT slice, and then each grid cell that has at least 50% lung in it is annotated according to a list of possible abnormalities, and this is done in a hierarchical order. So on the left, you can see the inspiratory scoring items. First, you check whether a grid box is filled with atelectasis or consolidation. If not, you go on to the second item, which is bronchiectasis with mucus plugging, and so on, until in the end, if there's only healthy parenchyma in the grid box, you will score that. Furthermore, if an exploratory CT scan was available, low attenuation regions were scored. The outcome methods of the best CT method are as following. First of all, the component score, which you just saw on the other slide. They are expressed in as total percentage of lung volume. And furthermore, we calculated the following composite scores. First of all, total bronchiectasis, which is a sum of component bronchiectasis and with mucus plugging and without mucus plugging. Secondly, we calculated total mucus plugging, which is a sum of bronchiectasis with mucus plugging and solely mucus plugging. Furthermore, we calculated airway disease, the combination of bronchiectasis, mucus plugging, and airway wall thickening. And finally, the total disease score, which is the sum of all abnormalities, except, of course, the healthy airways and the healthy parenchyma. Then the Hartman method. This is a CT score. It's based on the Brody score, and it's semi-quantitative. And it was developed to score chest CT scans for patients with common variable immunodeficiency disease. And with this method, each lobe is assessed, including the lingula. Lingula is a separate lobe, and the present and extent of the following abnormalities is scored basically by eyeballing. And it's a very extensive method because the observer has to assess in total 157 scoring items per CT scan. So it's quite extensive. The total Hartman score for bronchiectasis, total mucus plugging, and airway disease score, and the total disease score were also calculated by combining these scores, as was done with the best CT method. Then the intra-branch tapering. In healthy lungs, airway taper along the generations, so they do get smaller and higher generations, as is shown on the image on the left, image A. However, in bronchiectasis, there's a reduced tapering of the airways, which is shown in the middle image, image B. And this principle is used to calculate intra-branch tapering measurements. And what is done is the following. On a CT scan, the center line, so the middle of each airway, is manually drawn by an observer. And then next, the computer makes 3D segmentations of the airway lumen around this center line. And you can do that every 5 millimeters, as is shown on the image on the right. And then calculate the amount of tapering within each airways and between each airway. For this study, we use the intra-branch tapering, so the tapering within each airway, as is shown on the figure. Then the results of our study. This flowchart shows the inclusion of the CT scans. In total, 107 patients were included in the IBES study, and we received CT scans of 99 patients. Then, unfortunately, we had to exclude some CTs because they were not volumetric or incomplete, for example. And finally, we were able to use 84 scans for the analysis. And only 4 exploratory CT scans met the inclusion criteria. The patient demographics are shown on the right. The median age was 66 years and 64% of the patients were female. The median FEV1 was 59%, and the median BSI of the study patients was 10, which is quite high. This image shows is a stacked bar chart, and it shows all the results of the best CT analysis method, and each bar represents one patient. Every component score, which is shown on the right, has a different color, and the scores are expressed in as percentage of the total lung volume, and they add up to 100%. The y-axis is a little bit adjusted, so to improve the visibility of the graph. And what you can see on this scan is, well, first of all, there's a considerable heterogeneity within the patients, and considerable heterogeneity also of abnormalities. And the amount of disease ranges from 2 to 44% of the lung volume. Furthermore, what is important to realize when you look at this graph is that the amount of airways on a CT scan can be very low. For example, when you look at the image mostly on the bar on the right, only 5% of the total lung volume is covered by airways. And in this patient, which has the lowest amount of disease, still 2% of the total lung volume is bronchiectasis. So that is between 25 and 30% that is bronchiectic. So even this patient still suffers from considerable, high amount of disease. And what is also interesting about this image is the finding that the amount of mucus plugging varies widely, but there's also a lot of patients that suffer from quite a lot of mucus plugging. This is interesting because as bronchiectasis is considered to be irreversible, mucus plugging is actually something that can be treated and which might resolve when using the right therapeutics. This slide shows the correlation between the best CT method, so the CT analysis method we developed, with the Hartmann scores, so the other CT analysis method. And it shows the bronchiectasis, the mucus plugging, the airway disease, and the total disease scores. As you can see, all scores were significantly correlated, quite strongly. And this is basically what we expect when you measure the same outcomes, but then with two different CT analysis methods. Then the next slide shows the intra-airway tapering. In total, we measured 22,000 airways, and the median number of airways per CT scan was about 262. And for the analysis, we used the median of the median intra-branch tapering. And this graph shows the relation between tapering and total bronchiectasis scores. And there was a small but significant correlation between tapering and bronchiectasis. Less tapering resulted in a higher total bronchiectasis score. Then the correlation between FEV1 and bronchiectasis and FEV1 and total disease score. On the left, the bronchiectasis versus FEV1, and there's a significant negative correlation. So lower FEV1 resulted in higher, or was correlated with a higher bronchiectasis score, and the same accounts for the total disease score. So lower FEV1 is also correlated with a higher amount of disease. Then we looked at the reproducibility of the best CT score. We calculated intra-class correlation coefficients, and we considered everything below 0.5 as poor, between 0.5 and 0.75 as moderate, between 75 and 9 as good, and above good as and above 0.9 as excellent. And for the composite scores, we found only a good or excellent scores. And for the BEST method, the ICC scores were good to excellent for all values, but except for the percentage bronchiectasis with mucus plugging, and it was poor for the airway wall thickening. We know from other studies that airway wall thickening is often very hard to reproduce between observers and within observers. Between observers, the scores were still good to excellent for bronchiectasis and mucus plugging, ground glass, healthy parenchyma, total bronchiectasis, and total mucus plugging and airway disease, and total disease, but moderate for bronchiectasis with mucus plugging, airway wall thickening, and poor for empyema, belay, and healthy airways. So to conclude, we found with our study that BEST-CT is a reproducible scoring method to quantify and phenotype B patients, especially when you use the composite scores. The method correlated well with the Hartmann method. It also correlated a little bit less, but still significantly, with the intra-branch tapering and with a FEV1 percentage predicted. However, it did not correlate with other clinical parameters. Importantly, considerable heterogeneity was found within between patients, and what is also an advantage of the BEST-CT method over the Hartmann method is that due to the fact that it's grid-based, it's potentially can be used for automated analysis, which implies or which can be used in much larger data sets, and for example the EMBARK registry. There are several limitations for the studies. First of all, we used a retrospective data set, and the acquisition was not standardized of the CT scan, so there was a variety in slice thickness and amount of slices and kernels. Furthermore, the time interval between CT scan and the other clinical parameters was well deferred, but the median time interval was about 14 months. So the fact that we did not find any correlation with the other clinical parameters is not a big surprise, because the CT scans were not made during the first day of the study visit. And furthermore, there was no golden standard to quantify bronchiectasis, so we didn't have a golden standard to compare our BEST-CT outcomes with. Therefore, we used several outcomes, such as the Hartmann and the tapering and the clinical parameters. And finally, only 4 exploratory CT scans were included, and we consider this amount too small to perform any correlation analysis on this outcome. However, we do know from cystic fibrosis studies that low attenuation is often a robust imaging parameter. So in future studies, we would suggest to include this outcome as well. Thank you for your attention. If there are any questions, I'm obviously happy to answer them after the sessions.
Thank you, Jennifer. Thank you, Jennifer, for this thought-provoking, excellent talk. And while you were performing, there were several questions popping in, and we will have the opportunity to pick them up later in the course. So now it's a pleasure to introduce Ken Olivier. He is a senior clinician in chief of the Pulmonary Clinical Medicine section at the National Heart, Lung, and Blood Institute in Bethesda, Maryland. He's lead author of several key publications in the field of bronchiectasis and NTM, and actually a recognized player in the game of cracking the nut why environmental bacteria infect the one patient but not the other. And I'm looking to your talk, forward to your talk, Ken. Thank you.
Thank you very much. It's a pleasure to be able to speak today. I'd like to thank the conference organizers and the session chairs for this opportunity. What I'd like to do today is run through some thoughts on the association between genetics and bronchiectasis. I do have a few disclosures to list here. And then when we think of the strength of genetic associations, there are categories that we can consider. There are Mendelian associated genetic diseases, such as cystic fibrosis, alpha-1 antitrypsin, primary ciliary dyskinesia, and primary immune deficiency. There can also be risk alleles associated with, quote, idiopathic bronchiectasis, which is frequently associated with non-tuberculous mycobacteria. And those might include CFTR, and in this group, some studies have shown associations in a third to a half of patients in this category of disease, associations with SERPIN-A1, the gene responsible for alpha-1 antitrypsin deficiency, in a number of the cilia genes, heritable connective tissue disease genes, and immune-related genes. And then there's a category of bronchiectasis that includes allergic or autoimmune related diseases where the genetic risk may be less clear, at least in terms of how they relate to bronchiectasis in those conditions. And those might include diseases such as ABPA, rheumatoid arthritis, and inflammatory bowel disease. So what I'd like to do is go through a few representative cases that demonstrate the complexity of these genetic relationships. And the first is a 61-year-old woman with bronchiectasis, non-tuberculous mycobacteria, and alpha-1 antitrypsin deficiency. We initially saw her around age 48 when she presented with M. avium complex and M. abscessus. She had lower lobe predominant bronchiectasis seen in the circle here, a bit milder than her current CT. And her FEV1 was in the normal range at the time. She had no evidence of bronchiectasis on her scan, and she's had a gradual decline in her lung function over the course of years. Her genetic and other analyses revealed a heterozygous delta F508 mutation in CFTR, and her alpha-1 antitrypsin levels were low at 24. Her protease inhibitor phenotype was ZZ, and she had homozygous missense mutations in SERPIN-A1. And so with that genetic association, a couple of questions might come up. Could she benefit, for example, from CFTR modulators having a single allele involved? We know in patients with cystic fibrosis that these drugs have resulted in marked reduction in bronchial wall inflammation, and there may be some benefit in terms of effect on chronic infections. It's not yet known whether improvement in the control of MAC or M. abscessus can be seen, but it may be that the symptoms and disease associated characteristics related with these infections may be lessened under the influence of these drugs. Whether increasing the amount of CFTR in someone who has a single functional allele can be helpful is unknown, but the question remains. And then secondly, could she benefit from alpha-1 antitrypsin augmentation? This was considered at the time of her initial diagnosis, but it was felt with her normal lung function, no evidence of emphysema that this was not indicated. With her current lung function, while she still has no emphysema, I think this is being more strongly considered. So the relationship between alpha-1 antitrypsin and bronchiectasis is such that bronchiectasis has been described anywhere from 2 to 43% of patients who are PIZZ homozygous, so severe related disease. And in a study out of Colorado, severe alleles in alpha-1 antitrypsin, MS, MZ, and ZZ in a rapid grower NTM lung disease cohort were about 2.5 times greater than the frequency of these alleles in the general population, suggesting that this enhancement of abnormalities in alpha-1 antitrypsin related genetics might lend to predisposition to mycobacterial disease. Further, this study showed that alpha-1 antitrypsin reduced M. abscessus infection of human monocyte-derived macrophages with the thought that this may be requisite for establishment of chronic infection. And in these patients, if they had normal production of alpha-1 antitrypsin, it might lessen the opportunity for this to occur. The next case is a 31-year-old consanguineous female with bronchiectasis and respiratory failure. This is a publication out of the literature from the Mass General. And this patient had presented for lung transplantation with evidence of very severe bronchiectasis and cavitary lung disease. This patient had a prior history of neonatal respiratory distress, a finding that's common to patients with primary ciliary dyskinesia, and it had a history of recurrent sinopulmonary infections. However, the typical diagnostic tests that we now use for PCD, including things such as nasal nitric oxide, transmission electron microscopy examination of her cilia ultrastructure, and high-speed video microscopy were all normal. She had had cystic fibrosis and primary immune deficiency ruled out by clinical and genetic testing, and she had similar findings, though lesser in severity, in 2 of her siblings. She was subsequently found to have homozygous intronic splice site mutations in a novel gene called NEC10, which is thought to regulate ciliary growth and function, but does not affect ciliary ultrastructure as evidenced by the normal cross-sectional electron microscopic examination of her cilia showing the typical 9+2 arrangement. There have been 2 recently published guidelines to try to help some with what has traditionally been a confusing disease to diagnose. And I've compared and contrasted them in this table, and there are similarities and differences. They both start out in their algorithmic description with clinical criteria. These aren't specifically specified in the ERS document. The ATS document calls for 2 of 4 key clinical features: neonatal respiratory distress, laterality defects, and early onset purulent drainage from the nose and wet cough. Nasal nitric oxide is considered in both. In the European Respiratory Society document, it's considered along with high-speed video analysis. In the North American document, it specifies a criteria of less than 70 nanoliters per minute on 2 occasions greater than 2 months apart, having excluded cystic fibrosis. The high-speed video analysis is utilized, and noted that several centers in Europe employ this with high expertise. In the North American document, it was felt that high inter-rater variability and false positives and negatives limited its use. The use of air-liquid interface cell culture has been noted to improve the accuracy of high-speed video analysis and electron microscopy, but it's not mentioned in the ATS document. Immunofluorescence wasn't included in either document, but I think is becoming more popular, especially as more centers become proficient with it. And I think in most papers that are now describing newly discovered genes or associated genes, this plays a prominent role in demonstrating the presence or absence of the associated protein. Electron microscopy in both documents was considered useful in confirming the diagnosis, but we now know that there are multiple genes, such as the case I described, that result in normal appearance under electron microscopy, and it limits the sensitivity for diagnosis. And then finally, genotyping in both documents is also noted useful to confirm the diagnosis, but since we've not yet finished discovering genes, it does have limited sensitivity, though there are commercial vendors who are keeping up fairly well with genetic discovery, and the use of these panels can be useful in making the diagnosis. The next case demonstrates some of the overlap between PCD and other conditions. This is a 38-year-old male with bronchiectasis and recurring respiratory infections. He had lower lobe predominant bronchiectasis as demonstrated on his chest X-ray, and he presented to us for evaluation of primary ciliary dyskinesia at age 26 with a history of chronic otitis media sinusitis, and recurring bronchitis and bronchiectasis that had begun since age 8. His nasal nitric oxide was clearly in the diagnostic range at 13 nanoliters per minute. He had video microscopy, which suggested dyskinetic cilia, and transmission electron microscopy of the cilia was normal. He had quantitated immunoglobulins that were normal, and a CBC with differential of his white cell count was also normal. At age 35, he represented in referral for evaluation of 3 distinct lymphomas in his lung, mediastinum, colon, and colon that were refractory to prior treatment, and he had been refused conventional bone marrow transplant. About that same time, we got results from whole exome sequencing back on him that showed downstream stop mutation in IL-2RG. This is an X-linked gene associated with severe combined immunodeficiency. His mutation was far into the 5' end of the gene that resulted in near normal protein production, but not normal enough to stop one of the concerning complications of many of these primary immunodeficiencies of development of malignancies. And this is just a graph showing some data from a large multi-center study of the Genetic Disorders of Mucociliary Clearance Consortium in North America, where these patients were referred for the specific diagnosis or exclusion of primary ciliary dyskinesia, which was confirmed in about half of the patients. But in the remainder, a number of them ended up having conditions that overlap quite a bit clinically with things like otitis media, sinusitis, bronchiectasis, recurring needs for antibiotics and hospitalizations. And even some of them had nasal nitric oxide levels that were in the diagnostic range, but ended up having genetic mutations in genes associated with known primary immunodeficiencies, showing the considerable overlap between these 2 conditions. And to sort of break this down a bit more, primary ciliary dyskinesia is associated with defects now in about 50 genes that all result in a similar clinical phenotype. It is a rare genetically heterogeneous recessive disorder. In the US, it occurs in about 1 of every 15 to 30,000 births, or about 15,000 people. And it reflects abnormalities in ciliary biogenesis, structure, or function, and is manifested in organs where cilia play an important role. This includes neonatal respiratory distress, chronic year-round daily wet cough and nasal congestion that starts in the first 6 months of life, that's seen in about 90% of the patients in our multi-site study. And about 50% have laterality defects, either situs inversus or heterotaxy, where there's partial abnormal rotation. They also have age-related bronchiectasis at a very high prevalence, recurrent otitis media with frequent hearing loss, and reduced fertility in nearly all males. If you compare that to signs of primary immunodeficiency, and this is taken from the US Center for Disease Control's, their Genomics and Precision Health website, there's more frequent or repeated infections such as ear, sinus, and lung, and those are areas that overlap considerably with primary ciliary dyskinesia. In addition, there's also meningitis, skin infections, and thrush that is seen. And they further note that compared to most people, the infections last longer, don't respond to antibiotics or require IV antibiotics, are more severe or require hospitalization, things like sepsis or abscesses, and a higher frequency of opportunistic infections such as non-tuberculous mycobacteria. Things that overlap quite a bit with primary ciliary dyskinesia. So if you look at the genetic associations with primary immunodeficiency, there are defects in over 400 genes that cause these diseases, and they're categorized into multiple categories that include things like combined immunodeficiencies, antibody deficiencies, phagocytic dysfunction, and complement deficiencies. These are autosomal recessive or dominant or X-linked. The same gene can have both loss of function and gain of function with very different phenotypes. And the phenotypes in general can be very diverse, even within the same disorder or the same gene. And subjects with primary immunodeficiencies typically develop suppurative respiratory disease a bit later than PCD, though that's not always the case. And their symptoms can certainly overlap considerably that include recurring upper and lower respiratory infections. To try to sort out this overlap quite a bit more and maybe shed some light on clinical differences that are a bit more subtle, there is a newly starting study that is part of the rare disease research efforts of the National Institutes of Health and part of the Genetic Disorders and Mucociliary Clearance Consortium, which includes sites both in the US and Canada. And the goal here is to define genetic etiology of patients presenting with suppurative lung disease in children and adults. The primary objective is to utilize a systematic approach to identify characteristics that can distinguish PCD from PID and to take advantage of collaborations between pulmonologists and immunologists at each of the sites to perform the diagnostic and phenotypic studies and the use of genetic test panels for both PID and PCD with whole exome sequencing as necessary to identify the known or novel molecular etiologies, and then to compare these two for the prevalence and age-related severity of key clinical features that might allow refining the clinical diagnoses a bit more. The entry criteria, the age limitation starting at age 5 is to try to exclude those patients with severe immunodeficiencies diagnosed soon after birth. And the upper limit of 45 is to try to exclude many patients that might have idiopathic bronchiectasis or associated NTM disease, where we would be looking more for genetic risk alleles rather than Mendelian transmitted genetic disorders. The major clinical criteria is bronchiectasis in greater than 1 lobe. There are minor lung criteria that are required if there's no bronchiectasis, things such as neonatal respiratory distress, year-round wet cough, recurring bronchitis, pneumonia, or pulmonary NTM. And then there are minor other clinical criteria that includes things like upper airway involvement and low nasal nitric oxide. The exclusion criteria are aimed primarily at excluding patients with known causes of bronchiectasis or confirmed genetic diagnoses, and to exclude those patients with no standard evaluation for their suppurative respiratory tract disease. There are other efforts that we have in place at the NIH focus more on identifying genetic risk alleles rather than Mendelian transmitted disorders. And this is in association with the National Institute of Allergy and Infectious Diseases, where they have a centralized sequencing initiative that basically all patients enrolled on their protocols, including our natural history bronchiectasis protocol cohort, are eligible to be enrolled. They have specimens collected. They're analyzed, sequenced and analyzed by a molecular geneticist in concert with the research teams. And then a clinical report is generated and placed in the patient's record. And the research information is used for further study by the investigating groups. These samples are all linked on a website that is used by all of the investigators. So it gets around some of the stovepiping that can occur where similar genes are being studied in adjacent labs without people knowing about this overlap. We've currently enrolled 234 patients in a longitudinal cohort with detailed phenotyping. 195 of those have sequencing results available that have resulted in 14 confirmed genetic diagnoses of PCD, 7 genetic diagnoses of CF, 3 of alpha-1 antitrypsin deficiency, and 7 other relevant genetic diagnoses, many of whom had primary immunodeficiencies. The preliminary results looking at those without genetic diagnoses show very strong association with predicted protein altering variants in the 4 previously recognized categories of CF, PCD, heritable connective tissue disorders, and immune-related genes. And our future plans are to update the cohort risk allele association analysis using newer and improved databases and more complete coverage with more modern sequencing techniques. And we've begun exploring whole genome sequencing in this population. So the ultimate goal of this, I think, is to be able to categorize patients that could potentially lead to treatment modifications or improvement. And this is from a paper that several of us published in Lancet a couple of years ago, where it looked at breaking these risk alleles down into cilia-related variants that might take advantage of drugs that could enhance ciliary function, immune variants that could take advantage of multiple biologics that are targeting immune pathways, CF-related variants where the CFTR modulators might be effective, and connective tissue disease variants where some of the treatments that are being applied to those disorders might have a role in bronchiectasis patients as well. So I'd like to end there and thank people both in my lab, Kevin Finley, my cohort, my colleague in our cohort study, Kelly Kumar, who helps with our genetic analyses, and then collaborators at the NIH, Steve Holland's lab with the genetics, and in our consortium that is run by Mike Knowles at the University of North Carolina. And I thank you again for this opportunity to present and look forward to the question session.
So thank you very much indeed, Ken, for an excellent talk. And now we're able to move on to the discussion section that Felix and myself will moderate. We've got several questions already that people have typed in, which we'll get to in a minute. Before we start, Ken, that last Venn diagram that you showed is something that I've stolen for many of my talks on NTM as well. I see that now you've changed the middle bit instead of just saying NTM to NTM and bronchiectasis. Were the findings from your exome sequencing study for bronchiectasis pretty similar then than your previous published work on NTM as well?
I think there's considerable overlap there, and you have to consider that many of the patients that we see are skewed toward bronchiectasis, and I think that's an overall theme in the US in general, at least with regard to things like our bronchiectasis research consortium. So maybe it's a bit speculative to enlarge that to other patients with idiopathic bronchiectasis without NTM, but I think in terms of our genetic analysis, we are seeing similar risk allele assessments in those patients as well. It's just the numbers aren't as large.
Okay, great. Felix, do you want to ask anything, or should we just move to the written questions?
I think we should move to the questions. I would like to welcome Professor Guan from China. He has, in the meantime, joined the round. You're in China, actually?
Yes, exactly, Felix. Yeah, how are you?
Fantastic. We have really a world conference. It's wonderful. Yeah, there's one more or less comment from Harm Tiddens from Rotterdam regarding the talk on lung function. He pointed out that regarding LCI, there's more correlation with this forced expiratory flow values, 25 to 75, in particular FEF 75. It's rather a comment than a real question. Do you agree?
Yes, exactly. Exactly, I'll have to acknowledge that the idea of comparing the differences between the MMEF or FEF 25 to 75 and the LCI mainly stem from the research question that because both the parameters can reflect somehow the magnitude of ventilatory heterogeneity, but the effect of LCI measurement has already been well proven, but less remains clear for the MMEF. But it should also be recognized that according to a standardization paper published in the ERJ previously, that MMEF should be regarded as a less effective parameter in reflecting the small airway disorder. But nonetheless, many of the researchers might still might think that the parameter along with the MEF 25 or MEF 75 might reflect the small airway function. So the main point for the previously published paper reflected that the MMEF might not accurately reflect the small airway disorder, because it is usually abnormal even in some of the patients who have apparently normal FUN levels. And more importantly, the predicted values for these so-called small airway parameters will usually lie within extremely low values that might have already dampened the clinical interpretation of findings. But our previous point was that in some of the resource-constrained areas, so MMEF to some of the extent might serve as a surrogate marker instead of the LCI for the measurement of ventilatory heterogeneity in adults with bronchiectasis. Thank you.
Thank you for your answer. And I agree, in clinical practice, my feeling is that we still do not pay enough attention to these parameters reflecting small airways, and they are much easier to access compared to LCI. So I'll come back to that question later. So there's another question from the chat that is addressing Stefano's talk. Stefano, do you have any plan to validate usefulness of candidate biomarkers such as neutrophil elastase and desmosine in animal models? So there's a need to establish an animal model. This is a very hard question.
Well, this has been one of the key questions in the bronchiectasis research, Felix. We all know that we cannot produce reliable animal model for this disease where we can test biomarkers, drugs, and complications. So I think that as far as I know, there have been some publication as a poster in Congress about animal model in bronchiectasis, but no, really, I'm not aware about any good animal model where we can test this. Mainly because bronchiectasis as a disease is really heterogeneous, and it's difficult to define a single animal model. And the pathophysiology is strictly related to the etiology in most of the cases, as Professor Olivier just showed us. So really, really difficult. I don't think we have a solution for that.
And then what I think it makes it difficult in an animal model is also the time course. So usually bronchiectasis, even in patients with genetic disorder, develop over years and decades. And if you want to have an animal developing bronchiectasis in your lab, you really need to do something artificial with that kind of animal. So it's the time course and the history of the patient that make it particularly demanding. So let's move to the next question. Michael,
should I ask those? So there's three for you, Jennifer. Two of them are quite similar. They relate to how the best CT composite measure compares to other scores. So the Brix, Baller, Brody, and Reif are mentioned.
Yeah. So in our study, we choose to compare the outcomes with the Hartman method, but the Hartman method is actually based on the Brody. So the assessment of bronchiectasis is similar to that of the Brody score. And I think the Reif method was also mentioned, the Brix. Well, what all these methods share is that they are all based on eyeballing, and an observer looks at either each lobe or each segment of the lung and assesses the amount of bronchiectasis that is seen. And often this is done, scores are given between 1 or 3 regarding the amount of widening of the airway or the amount of the segment or lobe of the lung that is diseased. But these scores are, I think they are comparable. And we chose for the Hartman based on the Brody, but we could have used the Reef score or any other score. I think what is most important is that the best CT is different from those because it's grade-based, and therefore it could be used for automated analysis. The Pragma CF score for cystic fibrosis is also grade-based, and currently this score is almost it's almost possible to analyze CT scans automatically. This will be done in the beginning of next year for a lot of CT scans, and the results will be shared by then.
Great. And just very briefly, while I've got you concentrating, the other question directed to you by Dr. Steinberg was that she was surprised that the number of patients with was few. Surprising number of patients with very few bronchiectasis on the CT scan, but they still had Pseudomonas infection. Can you hypothesize why?
Yeah,
I think suggested perhaps chronic bacterial bronchitis.
Yeah, so the CT method, the results are expressed as percentage of total lung volume. So I agree that a score of 2% of bronchiectasis, it doesn't sound like that is a lot, but when you compare it with the total amount of airways that is visible in the lung, it's actually still quite high. So while the percentages are low, still the amount of disease in all patients was considerably high.
Okay, thank you. Felix, do you want to ask the last two?
Yes, there's one question from Ricardo José directed to Professor Olivier. So when do you suggest whole genome sequencing, or I think whole exome sequencing? This is something that we actually at our institution perform for the last 3 years as a routine of clinical care, and the results are pretty much the same that you have. We are getting confused by genes never heard before. Me as a pulmonologist, I have the opportunity to learn, but I also have the task to recommend functional workup for these genes in order to reason the significance. So when do you tell the indication to trigger this genetic testing?
Well, it's an excellent question. I think getting the sequencing is the easy part. And as you mentioned, figuring out what to do with it is much more difficult. And especially, with conditions like primary ciliary dyskinesia, where, there are 50 different genes, they can have different sort of biomarker presentations in terms of things like nasal nitric oxide or ciliary ultrastructure abnormalities. It's even more confusing with primary immunodeficiency. There's just hundreds of these genetic associations. And so I think if you're going to launch into this, you need to be equipped with an immunologist and a molecular geneticist in your back pocket. And in terms of trying to figure out the significance of a number of these missense mutations or missense variants, if they're not published in the literature, for conditions like PCD, it can take quite a bit of effort to do that. A lot of what we're doing when we come up with these not previously described missense variants are relying on our Genetic Disorders and Mucociliary Clearance Consortium and the enlarging genetic database associated with that to help guide us, as well as research teams at UNC and other places who can look at more detailed assessment of the presence or absence of protein and do functional studies associated with that. Similarly, for the primary immune deficiencies, having someone that can parse through those and figure out which ones need to have further immunologic investigation can be really helpful. Otherwise, you are spending a large amount of your time kind of chasing your tail, trying to figure out how to analyze all these genes. And so I would just put a plug in for, as a stopgap measure, if you don't have those kind of resources, the use of these genetic panels, which are increasingly available and are excellent for primary immune deficiencies and are getting better for primary ciliary dyskinesia. The advantage there is that these vendors come with their own molecular geneticists who can help you parse through that and give a much more interpretable report than you'll get back when you receive raw information back from whole exome sequencing or whole genome sequencing.
Yeah, these results really make me weird. And I think that given what you have shown, that the study you presented is really best at your hands to answer the questions, giving the power and the staff and the forces you have, and congratulations to that setup. This is really marvelous. The last question, and it's a wonderful person asking this question, and it's a wonderful question. It's from Thomas Ruddy. He's patient from Germany, and actually Thomas Ruddy, that brings me back to the motto we had with the first bronchiectasis World Conference, joining forces for a breakthrough in bronchiectasis. And we came up with the slogan. Thomas came up to me and asked me, oh, I didn't know that you cracked the nut already. And I said, no, Thomas, it's stay calm, and we're just joining our efforts. And so he's asking, should we as bronchiectasis patients realize that it's only a matter of time before enough evidence of genetic causes of bronchiectasis are discovered? So my suggestion is, as the four of you are sitting there, maybe let me ask the question like that. All the four speakers addressed, what is your recommendation to join the best combination of functional assessments, biomarkers, radiological assessments, and genetics, and in particular the combination of radiological assessment and genetics actually have a very high, in my point of view, have a very high likelihood of defining the genetic disease. So if you all four stick together your heads, would it be possible to really are we already there to resolve every idiopathic bronchiectasis case? So who's ever first?
Well, if I may start, what I think when we look at our results, we see a lot of heterogeneity within patients, and this implies that there might be several types of bronchiectasis, which could obviously be studied with other parameters such as the genetics. So I would be enthusiastic to combine these outcomes indeed.
Okay.
I can make a comment. I think many of us look at asthma as sort of a goal to get to. It started out with looking at studies of a variety of treatments that look promising in well-controlled phase 2 trials, failed in phase 3 trials, and I think as asthma investigators got smarter and learned how to endotype or categorize or put these patients in bins, that has then led to more success therapeutically. I think many of us are clinicians, not geneticists, and I think we're still reliant on our clinical findings when patients present. If they present with a family history of multiple patients with bronchiectasis, they have early onset of their upper and lower airway disease. They have other findings that might suggest a genetic etiology. I think that pursuing that more aggressively can be helpful. I think many of us are now comfortable with diagnosing cystic fibrosis genetically. Granted, it's a bit more simple, even though it's quite complex, but there's a single gene to look at, and we get nice reports back that pretty much say positive or negative when they come back. We're a long ways away from that for many of the other genetic disorders associated with bronchiectasis. So I think it's going to take a bit more work, but I think the tools are there. It's waiting for the research to be done to figure out how to take a very complex situation now and refine this a bit to where it's usable in the hands of clinicians to be able to parse these out.
Would this be an issue for artificial intelligence? So feeding CT data in genetically defined patient cases and programming and building up models for predicting disease course? Perhaps this was, at least was one of the last comment that just popped in from Harm Tiddens from Rotterdam. So there are no more questions, and I'd like to thank you for the wonderful discussion and hand over to Michael.
So yeah, just left to me to thank all the speakers for their excellent talks, and I thought it hung together really well as a session actually, and thanks for all the audience as well for their participation, which made Felix and my job a lot easier. And it's up to me now to invite you all to join the virtual cocktail. I'm not completely sure exactly what's going to happen there, nor what we're meant to do, but I think you're meant to all stay here, and we'll suddenly see 300 people on little screens or something. Well, I look forward to seeing what happens, but thanks again for your participation here.
Thank you.
So now is the time for the virtual cocktail slide. This is very strange, no? But I think it's going to substitute a real cocktail. And it is my pleasure again to say hello to all of you, to everyone. I think it is unbelievable that we can be here in this World Congress of Bronchiectasis and Non-Tuberculous Mycobacteria. I enjoyed this morning's sessions, really. And it has been an effort, but we are here. We are here in this year of living dangerously, like the film. Eh? Have you seen the film? No. Yes? Okay.
Yes.
Yes,
we do. Yes.
So this is the second year of living dangerously. After the film. So what we have to do now is to show first the first piece of paper that says United We Care. Please show that. Those that you have and you keep it. Okay? The importance to be united, very important in this pandemic. Stefano, what is your piece of paper, please? You don't have it. Okay. And then now, secondly, intensively, very intensively. Jennifer, okay. Thank you very much. Very much. I think this is going to be for the eternity. Okay. And my best wishes for all of you. My best wishes of friendless, and thank you very much for your participations wherever you are now. Thanks a lot. Francesco, do you want to say something?
Just cheer.
Just cheer. Tim.
Salud.
Salud. This is wine. This is wine.
Salud. Salud.
Salud. Salud. Salud to everybody.
Salud. It's health. For us, it's health.
Okay, and my respect for the health workers, the frontline health workers that took care of the patients with COVID, and for scientists as well. They have done a very good effort this year, unbelievable. 47,000 papers published since the beginning of the pandemic. Unbelievable. So I think that we deserve some respect.
Yeah.
Thank you very much.
And working together again and again.
Enjoy the meeting.
Fantastic. Well
and
done.
your cat as well, Jacob.
Fantastic.
Fantastic. Thank you.
Bye.
Goodbye.
Thank you. Bye,
Thank
everybody.
you. Bye-bye.
Bye.
Together.
Together.